Welcome everyone who is just joining us. My name is Lauren. I am from the 3Rs Collaborative, and I'm excited to give you a quick intro to our webinar series today. You're going to hear from an exciting group of 3RC MPS companies. So I'll just dive in without further ado. So just a quick disclaimer, this webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussion of competitively sensitive subjects, including cost, prices, sales, product marketing, and other confidential information. In addition, neither the 3RC nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. So just to begin, I'm going to provide you with a quick introduction to the 3RC and this webinar series. So if you haven't heard of us before, you're joining us for the first time. This webinar series is initiated by our MPS initiative, which collaborates to accelerate adoption of MPS in scientific research. And our initiative seeks to increase adoption and regulatory acceptance of MPS in coordination with animal studies. Our members span across the scientific field, so you can see a number of diverse institutions and individuals from different levels of academia, government, pharma, and this is just our leadership board, but this spans also across our institutions and our member companies and individuals as well. As you can see, we have 42 institutions who are members of the 3RC MPS initiative. And this initiative has a few aims. One, to provide thought leadership and expert consensus, to facilitate appropriate discussion, collaboration, and sharing between developers, to create direct engagement between developers and regulatory agencies, as well as develop external partnerships. And finally, to create resources to facilitate increased engagement with developers. And we accomplish this via our three work streams. One is our regulatory, our technology expo, and our end user and education subgroup. So we're consistently engaging with end users and providing education. So I just wanted to point you specifically to our 2024 updates, where we joined Altex as a member. We've conducted quarterly workshops, and we've also talked at the MPS World Summit, SOT, and Eurox. You can see past presentations, as I mentioned, our past workshops on our presentation page. And in terms of our regulatory group, in 2024, I submitted a publication on perspectives on using MPS for regulatory applications, established a PPP with FDA CDER, as well as a cross-platform DILI project. And finally, for our technology group, we launched this hub in 2021, but in 2024, we continue to add disease areas and tissue filters. And we're also currently developing a publication hub, which we're really excited to hopefully launch in 2025. And this here is the link to our technology hub. If you have not visited it before, you definitely should go check it out and see all of the great companies that you'll hear from today. So I just wanted to remind everyone to stay tuned for topics and save the dates for our 2025 webinar schedule. We're really excited to continue this partnership with the IQ moving into the new year. A few reminders for this webinar. Please submit your questions using the Q&A box, ideally instead of the chat. That's just where I'll check first. And questions will be answered at the end of the webinar after each presenter as time permits. Feel free to email me if you have any questions about our MPS initiative or interested in becoming one of our sponsors. And I will now turn it over to David Kukla from the IQ MPS initiative.
Thank you, Lauren. Hi, everyone. My name is David Kukla, and I'm part of the IQ-MPS. Specifically, I serve on our strategic partnerships and communications team. So I just want to give a quick overview of the IQ-MPS. And the IQ-MPS is currently composed of 27 member companies, and we have representatives from drug safety, 3Rs, ADME, as well as PK/PD experts. And our leadership team is made up of Anya Kopek, Rhiannon David, Kim Homan, and Rhiannon Hardwick. But besides this leadership team, we have a steering committee, and this is made up of 2 individuals from every one of our 27 member companies. And then to the right, I'm just highlighting a little bit more about the IQ-MPS. We have over 150 active participants, which meet several times a year. Additionally, we have several publications and are currently working on several others soon to be published. Also, I want to guide everyone to our LinkedIn page that we recently set up. And you can use that to kind of keep up with all the IQ-MPS activities there. And the IQ is made up of 4 main goals. One, to serve as a thought leader for both MPS developers and stakeholder organizations in the industry implementation and qualification of MPS models. Also to provide a venue for appropriate cross-pharma collaboration and data sharing. To create focused engagement between industry and regulatory agencies on the current status and evolving field of MPS, as well as to develop external partnerships and collaborations to help enhance the inclusion of industry priorities. And with that, the IQ-MPS is made up of 5 affiliate work streams. There is the organotypic manuscripts team, where the goal is to publish manuscripts outlining organ-specific industry requirements for MPS platforms. Regulatory outreach, which identifies opportunities to engage with health authorities globally on topics related to MPS qualification. Also, there's the pilot project teams, and this team initiates near-term proof of concept studies for data sharing or prospective collaboration for qualifying MPS systems based on specific context of uses. Also, the strategic partnerships and communication team communicates industry perspectives and builds relationships with key MPS stakeholders to enhance the inclusion of industry priorities. And then finally, the landscape gap survey team conducts and analyzes surveys benchmarking how pharmaceutical companies are currently using MPS. So here's just an overview of our external partnerships over the past few years, starting back in 2018. But besides this, we do have several other activities, including cross-pharma collaboration and data sharing, regulatory agency engagement, as well as thought leadership. But some of the highlights from our external partnerships team, as Lauren mentioned, in 2021, we co-partnered with the 3Rs Collaborative and established an MOU, which allowed us to work together on webinars and workshops such as today. And in 2022 and 23, with the 3Rs Collaborative, we had 4 webinars each year on different MPS systems such as neuro, liver, blood-brain barrier, vascular, and GI. And then within 2024, we had 4 additional webinars on topics including immune models, neuromuscular and fibrosis models, developer perspectives on MPS and cell sourcing, and then finally this webinar on oncology models. And we're really excited to continue this effort in 2025. So right now we're working on kind of those topics. So we'll get back to everyone in early 2025 on that. But if you have any questions on the IQ-MPS, please feel free to reach out to me or check out our website that I have listed in the lower left corner, iqmps.org, to keep up with any of our activities. Thank you so much.
So I'll go ahead and get started again. Thank you so much for the invitation. Just before I begin, I wanted to give kind of a brief overview about ATCC. Founded in 1925, we are reaching our centennial here. We are a nonprofit organization that's headquartered in Manassas, Virginia, with an R&D center in Gaithersburg, Maryland. Now, for those of you not familiar, we are one of the largest and most diverse biological materials and information resource for cell biology, and we are often referred to as the gold standard for cell models. Now, we're also an innovative R&D company, and today I'm going to be talking to you about our Microphysiological Systems, or MPS program here at ATCC that's focused on developing new and advanced cell models to accelerate oncology drug discovery and screening. So the Microphysiological Systems program was established at ATCC to contribute to the scientific community around the biological pain points that are associated with adoption of MPS programs. The program currently has 13 working scientists across 4 main segments. The first segment that we are working on is our cell derivation unit, which is focused on primary cells. This team is responsible for isolating and preserving these cells, developing protocols in additional cell types, as well as characterization of these primary cells. Now, the StemCell team is also working on protocol development, checking the differentiation, functionality, and compatibility of these cells, including their long-term expansion. My focus today would be on the oncology models that belong to the 3D models segment in this pillar. We are developing and manufacturing and cryopreserving patient-derived cancer models, which I'll be focusing on in today's presentation. But lastly, the technology segment of this MPS program at ATCC is really, in this block, we are using multi-platforms that are commercially available in the MPS field with various levels of complexity to test our biological cells, including the cells from a cell derivation unit, media, and ECM, to support development of new models in the MPS field and access new cell functionality in these models. We often collaborate on these with our MPS developers that are on this call as well for new products that can benefit our end users. So the models that I'm going to talk about today are heavily focused on oncology. They belong to the Human Cancer Models Initiative, which is an international consortium that is tasked with generating 1,000 new patient-derived models or next-generation cancer models. Now, all of these models are well characterized and associated with a variety of clinical and molecular data. These are from diverse patient backgrounds. Some of the data metrics I'll be sharing with you in the next few slides. And these utilize advanced cell culture techniques, and our technology platform is integrating these models with our MPS systems. Now, HCMI founder and members are the National Cancer Institute and the Cancer Research UK primarily, with hub organoids and all of these institutions mentioned here as the founding members. We are accessioning new models from this consortium continuously, with new model deposits coming in from Stanford University just most recently. Now, we've been supporting this program since 2016, so we have quite a few models now in our collection, and our main role here is the authentication and quality control testing of these models, including protocol development and the scale-up and manufacturing of these models. We also contribute back to this HCMI consortium in the form of model information resource by providing models back, including data back, and the distribution of these models, we are the exclusive distributor through ATCC. We have 316 models now available. These are representing 28 different tissue types. 75% of them are organoid models that we are integrating with our MPS systems. 10% of them have shared donors, which means that we have models from both the primary tumor as well as the metastatic site from the same patient. And we release about 5 new models per month. Now, our focus is to prioritize releases, not only for integrating these systems with, commonly diagnosed cancers, but also serve the community in rare cancers. So we had our first head and neck cancer organoid model released, as well as the first desmoid tumor cell line that we released through the HCMI collaboration. Our production pipeline focuses on lung, breast, endometrial, ovarian, and all of these tissue types that we have within these next generation cancer models. Now, the derivation is all from primary patient tissues. The cancer model development centers develop these organoid models. They, once they're established, they're sent over to ATCC, who then reproducibly creates protocols for scale up of these models so that they can be distributed back to the global scientific community. There is a lot of advanced molecular characterization, some done here in-house at ATCCs and a lot of it done at the cancer model development centers. Most of the models at least have at minimum a 15x whole genome sequencing on the model itself, whole exome sequencing or RNA sequencing, and include DNA array, depending upon which cancer model development centers these models are being accessioned from. Now, we do also provide the clinical data sets associated with these models. We have standardized case reports forms as a part of the HCMI consortium that includes patient demographics, disease diagnosis, treatment and outcome information for these patients that are incredibly valuable for the use of these models in research. So far, the HCMI consortium has developed about 763 models total that we have in our collection. Out of those, we have launched 316 models. Now, as I've mentioned before, they contain rare adult and pediatric cancers as well, including rhabdomyosarcomas, leiomyosarcomas, Ewing sarcoma, and others. Now, the top 10 models are represented here. 80% of the total collection includes brain, pancreas, and colon, which are the most common cancer types that we have in our collection. Some more information on these models includes the clinical and demographic span of these models. 42% of our patient models are from female patients, 14% non-white, 23% have gone through neoadjuvant therapy, and 65% of these models have all of their therapeutic details available. So you can look at whether the patient went through neoadjuvant chemotherapy, what the outcome was, and if they've gone through standard chemotherapy, which really gives this the consortium an incredibly powerful, diverse model resource. Now, the age distribution of these models primarily is represented here for all of the models, but we are increasing our patient diversity and we are focusing on producing more models that are focused on pediatric and rare cancers. Now, one of the critical thing is that the patient-derived models from the HCMI are capturing the genotypes that's seen in actual patient populations. Now, the TCGA, the Cancer Genome Atlas, that was a landmark cancer genomic data set that contained over 20,000 primary tumor samples that provides a comprehensive molecular landscape of cancer. So in this slide, what we are showing here is that we've compared our HCMI models to the TCGA cohort to get the sense of representativeness that these models have. Now, these 316 models were all included in these analyses that we have expanded and are distributing currently in our collection. We see a high concordance ranging from 80% in colon cancer for cases that have mutations in canonical oncogenes that are widely studied, that are recognized and are being currently targeted with drug screening. We also see canonical mutations that are very representative in cancer development and progression across all our model types. P53, BRAF, NRAS mutations are all very well represented within this model base. Now, all of this information is available. So the model selection tools that we have developed with the NCI that allows researchers to purchase these models, look at what these organoid models are, what they represent in terms of their mutated genes. You can use missense mutations or any kind of mutations that you're looking for, particularly in these models, and you can filter down to that particular patient representative model and purchase that model through ATCC. Now, we're also doing some of the work here, in addition to that, of characterizing these models and then integrating them with our MPS platforms. We are also doing some screening work on these models that's going to be presented in AACR in 2025, where we are using these models, seeding them into plates, and then testing them for sensitivity to common chemotherapies, which are single agent chemotherapies. So on the left panel here, the data that I'm showing is showing targeting these 4 PDM models from pancreatic cancer organoids with 5-fluorouracil, cisplatin, and oxaliplatin. And as you can see, you see a high representation with exhibited a clear dose response curve with significant toxicities in all the models. And these are in all of the derived models, which is very important since the expansion of the models, we do lose some diversity in the heterogeneity of these models as we expand them. But when it comes to looking at it from an oncology perspective, they show extremely high concordance and responses to and toxicity to standard chemotherapies. Now, when we look at targeted chemotherapy, here we are looking at specifically targeting G12D mutations in these models. So 2 of our models here in the orange and blue, which is PDM36 and PDM37, each representative to a single patient, actually have the G12D mutations that we characterize them for. And as you can see that they exhibit that greater sensitivity compared to those models or patients from whom we have models that have the G12V mutation or the KRAS wild type. So this data, including the integration with our MPS platform of adding normal back into these models, will be available at AACR. Now, just very quickly, ATCC is also a resource for the information on the HCMI models. So from our HCMI landing page, you can search and filter among the currently available models that we have for distribution. From there, we can provide model and lot-specific information, including the QC testing results, model-specific protocols, model images, as well as links out to additional clinical and molecular characterization that might be available externally or internally through ATCC. Now, beyond simply providing the models, ATCC definitely wants to ensure that you can use them in the lab and they're going to be successful. So we recognize that some of these models are extremely challenging to work with, specifically the rare desmoid tumor lines are extremely challenging to adopt. So what we've done is we're sharing all of our protocols, including having specific growth kits designed for the expansion of these models. We have released these select companion reagents and growth factors kits that are specifically aimed at supporting the HCMI models, making that culture very easy, including sharing protocols on how to do some screening and integration of these models. Our website is also an incredible resource for educational information around the HCMI models, including video protocols, application notes, and suggestions for troubleshooting. We have still got a lot of work to do. We currently have a backlog of about 300 HCMI models that haven't entered our production pipeline yet. While our intent here is to make them available, we are asking the research community for their input and prioritization. We have made available a list of these unreleased models on our website, and the HCMI landing page will soon be updated and to show the unexpanded models that you can browse, select, and let us know to prioritize into our production queue to be made them available. Now you can, the feedback that we are looking for is you can filter by specific models, tissue genotypes, patient demographics that you're interested in, and we'll be able to take that into account. And you can contact us at hcmi@atcc.org. So this is my last slide, and I'll just end by highlighting a couple of additional resources where you can learn more about the patient-derived cancer models from HCMI at ATCC. The first is the HCMI landing page that we have on our website. The next two are resources that are not ATCC, but maintained exclusively by the National Cancer Institute and contains useful information about these models. The first is the HCMI searchable catalog, and this is where I was kind of alluding to, where you can filter these models down to primary site, the kind of mutation, somatic variant type you're looking for, and look at all of the clinical information that's available on these models. We also have all of the data for characterization available, and you can access that without purchasing the models directly through the NCI by creating an account at the National Cancer Center Genomic Data Commons portal, where all of the model-specific large cancer data sets are available. This includes raw whole genome sequencing, whole exome sequencing data files. Now, one of the key features of the HCMI is its commitment to its open access to researchers worldwide, so focusing and fostering collaborations. And I know this webinar is also a platform for us to bring to these models. And if there are any other partnerships that we can provide these models for, we'd be happy to have that discussion. Thank you so much again for inviting me.
Thanks so much, Yitzav. There is one question. We have time for one in the Q&A. The question is, are there matched PBMCs for the cancer organoids?
That's a great question, and we frequently get this question that, or have we, expanded and banked? There are few cancer model development centers that began doing that work very late. So in some of our new models, we do have some match PBMCs that are banked, and as soon as we start releasing them along with the organoid models, we'll provide that information on our HCMI landing page. But for the models that were accessioned and already produced, the 316 I talked about today, unfortunately, we do not have the matched patient PBMCs.
Great. Thank you so much, Yutsav.
Perfect. So I will be talking with you all here today about modeling CAR T-cell therapies and utilizing organ chips for that. So as a brief little background for this, so CAR T-cell therapies have grown a lot of traction in the past few years. We've seen that there's been a lot more uptick in wanting to do research and development within this space. And so CAR T-cells, it stands for chimeric antigen receptor T-cells. So these are engineered and modified T-cells to be able to then have a targeted tumor killing capacity for them, and to be able to hone that and direct that towards the specific therapy or the specific tumor of interest. Presently, there are 6 different FDA-approved CAR T-cell therapies, all that are primarily focused on blood cancers at this time. And then, though, in this later half of the year, there has been some more uptick in being able to evaluate for more additional applications too. However, blood cancers really represent around 10% or less of the total cancer cases and deaths for each year. And so as an effort to be able to then direct these and be able to provide a solution as a tool for researchers to be able to evaluate CAR T-cell therapies for solid tumors has been a continual challenge for researchers. And so we've been hoping to be able to look to address that here with this. So a little more background. So typically 2D cell models are what's really been utilized for research in the development of these CAR T therapies. And so really what they allow you to do is be able to evaluate really kind of, as we say, one piece of the puzzle at a time. So focusing primarily on killing. There are other assays such as migration and being able to look at some of these other components individually, but really that they are lacking in the holistic approach of being able to capture everything from A to Z within what happens with these CAR T cells, and being able to then address what happens. Because as we evaluate the capacity for being able to then see that these are able to getting to the tumor is obviously the really critical component, the rate limiting step. And so, okay, so if they can kill this CAR T or this tumor cell through these CAR Ts, that we often see though that there's just going to be a dropping off of the amount of CAR Ts that can actually reach that tumor. So needing to understand, how do we dose this? How do we correctly correspond what this is going to look like for being able to create a more effective therapy, and being able to understand, understanding the right affinity binding, et cetera, for these different therapies, and being able to then capture that so that there can be a better modeling and understanding for this. So we are a 2-channel microfluidic system. So what we have here is just a schematic of those 2 channels. So we have our membrane that is separated here in the middle. And so with that, it is a porous membrane. It's made out of PDMS primarily. And though we do have other chips and things that can also be utilized for these applications, where they also have...
Sorry to interrupt, but your slides aren't advancing.
They are not advancing, you're saying?
No, you're still on the first slide.
Okay. Let me see if I can end this. Let's see if I can go here. Is this better?
There we go. Yeah.
Okay, perfect. Sorry about that. So what we can see is that ultimately, so we are a 2-channel system, so we have a porous membrane. So in this case, what we initially developed this system on is our S1 chip. It's our very tried and true commercially available chip. But we also then have our now rigid chip with an R1 that then has a different membrane. It's also a bit thinner here. So that also then allows for a little bit more cell crosstalk and being able to still enable these applications for looking at immune cell recruitment and being able to understand that. So what we have is that overall what we add in our tumor cell line in this top channel. In this case, what we were developing was an A549 HER2 positive cell line in this top channel to be able to mimic non-small cell lung carcinoma. And in the bottom channel, we have lung microvascular endothelial cells. And so with this, we then also include a chemokine gradient. So we add in some tumor-relevant chemokines. So there's a list of about 7 or 8 different chemokines that we add into the top channel. We prime the surface of the endothelial cells, and this is to induce kind of a mimicked disease state. So being able to then use primarily TNF-alpha, but this can also then be adapted depending on the tumor model that is being modeled and what is most relevant. A lot of times, some people are also interested in IL-1 beta, and there are other possibilities around that as well. And so with that priming stimulus, what that then allows us to do is we use this, we then induce a high flow rate to be able to then also ramp up and increase shear forces. And so with that tuning, we have our CAR T cells that we then have inside of a buoyancy media. And as they flow along the bottom channel, they're able to first adhere to the endothelial cell surface. With that chemokine gradient, they are able to then migrate and be able to infiltrate into the tumor cells, being able to then see that migration, therefore also then seeing that target-specific killing happening as a result. And we monitor this typically for around 48 to 72 hours post-administration of the CAR T cells. So with that, so really what we wanted to do is develop a workflow for understanding solid tumor CAR T cell evaluation. So this kind of breaks down to 3 main categories. So first, of course, the target cells. So what are the cells that you're going to be utilizing? This can also be it's up to users if they want to ramp up the complexity. So if you're wanting to do simply a cell line, a cancer cell line in the top channel, you can. If you want to do things of generating spheroids or doing other things, being able to include cancer cells with healthy cells into that top channel, that's certainly up to the discretion of the researchers looking at that. And then, but in this case, what we were doing is HER2 positive A549 cells. And then also we were wanting to then using a non-targeted control for CD19 positive A549 cells. For CAR T cells, so we're using second generation commercially available HER2 or CD19 CAR T cells that we procured from ProMab. And then those were then administered via the tissue-specific vascular channel with then shear forces, of course, induced through that high flow rate. And so all of these are commercially available frozen vials. So these are things that our users and customers are able to then procure and be able to then repeat this and reevaluate this for themselves. But then also there can also be the inclusion of co-therapeutics. So of course, IL-2 is a very commonly understood looking at this for preconditioning and as well as adding this in to enhance the killing capacity for the CAR T cells, but also other co-therapeutics are certainly available to be able to be utilized according to your needs and research interests. So diving into a little bit more then of the data, so being able to then first evaluating what can be utilized on organ chips and how can this really look like to our researchers. And so with this, what we can see is that we're able to, after that administration, we can actually stain these immune cells prior to administration with Cell Tracker. It's a very simple and effective way to be able to monitor the CAR T cells and where they are within the chip system. And so here we're able to then flow those through, and then using a high content imager, we're able to then quantify the number of CAR T cells that are present on the chip. And so we can separate those out based around the space and the size of the channels, as well as the membrane space, and be able to then quantify and being able to then take these 3D confocal imaging to be able to segment and quantify those immune cells that are attached, and in this case being the CAR T cells that we're able to then see migrating into that top channel. So with that quantification, we can see that the HER2 positive CAR T cells with the priming stimulus comparatively to without that priming stimulus. And so priming stimulus, just for reference, that's again that chemokine gradient in that top channel, as well as that TNF-alpha, in this case, in that bottom channel to prime and upregulate those cell adhesion molecules on the endothelial cell surface. And so we can then be able to see that the CAR T cells greatly increase in their migratory and infiltration capacity into that top channel especially. And so we're able to separate out that we see quite a few, yes, attached to the endothelial cells, but with this priming stimulus, they really migrate up into that top channel. And we see a significant quantification there of those. Comparatively, with our CD19 as our negative control CAR T line, we do see a little bit of some CAR T cells that are then present, but we'll get into that a little bit more in this next slide. So here what we are able to then see is really actually that visualization again of those CAR Ts. So with the CD19, what we're actually stating here is for also then caspase 3-7 cleavage. So we're looking for cell death and apoptosis. And we can see that we see some presence of these CD19 CAR Ts on the chip. However, we're not seeing really any signal for caspase signaling taking place. So we're seeing that, okay, yes, they are present on chips. Some did attach, which is to be expected. However, we're not seeing then an effect and an activation of those CAR T cells to be able to showing any killing signal there. Like inversely, we see that with the HER2 CAR T cells that we do see then this killing, very robust signal that we're seeing here, which is then quantified on that right-hand side. And so we can look at the total number of caspase objects that we're able to then analyze on those images and be able to reference those. And so what we can see then is being able to then separate this out between a negative control CAR T and a positive CAR that we can be able to then disseminate between both of those and their killing capacity. And this is something that we are also, you'd be able to then adjust this towards your needs. So if you had something where you were wanting to be able to understand different killing capacities, certainly of different CARs, and maybe you have some different targets that you would then kind of be able to then modify on those, you'd be able to then kind of rank order those and be able to evaluate that on chip. But in addition to this, there's also, of course, the effluent. So from having a continuously flowing system, we're able to then collect those effluent at regular intervals. So in this case, we're looking at the 48-hour interval. And so what we can see is that with the presence of no CAR T cells, we're not seeing any of these cytokine release or any of these markers. However, that we do see specifically with the top channel effluent that we can see the presence of both degranulation as well as cytokine release from looking at perforin and granzyme B on that degranulation side and looking at interferon gamma and IL-2 as well then in the presence of those CAR Ts and with the HER2 specifically. So we do see a little bit of elevation in IL-2 even without the priming stimulus, but we do see some larger error bars there that are present. However, we see very nicely a very strong and robust signal for that with the priming signal with those HER2 CAR Ts that is present, and that also then correlates with the caspase 3-7 signal and being able to then measure that tumor target killing efficiency and being able to look at that. Likewise, so kind of understanding things in part where we can then look at those at time dependency. And so we can look at time dependent killing activity. So this is being able to be shown by first, of course, looking at degranulation, as I mentioned, granzyme B and perforin, but also cytokine release. So we were kind of really focusing on these main two cytokines and main two degranulation markers that we found to be very, very strong and be able to be very easy and readily available and used by customers. And so we then quantified those and be able to look at those presence overall. So we see that very little is present at the T0 mark. We see initially, a peak with some IL-2 at the 24-hour mark, and likewise with perforin. And we see very nicely an increase in signal within that 48 hours that coincides with the effect in which seeing with increased caspase signal and being able to then correlate that as needed. So this really is able to inform how these CAR designs with different signaling domains or with different co-therapeutics might be able to alter the time dependency in that killing signature in ways that could inform mechanism of action. So, but being able to also understand looking at immune cell extraction and phenotyping. So within this workflow, we're able to then remove those recruited immune cells, in this case being, of course, the CAR T cells, being able to then perform the immunophenotyping. So on the left-hand side, what we're looking at is first flow cytometry analysis of the recruited immune cells for cell surface PD-1. So that's, of course, a marker for immune cell activation and being able to see antigen-specific results that correlate with that recruitment and the killing that we just saw previously through looking at effluent and looking at fixed chips. And so with this, we can then also look on the right-hand side, though, and being able to use this, use the effluent from those same chips, being able to then evaluate secreted PD-1, being able to look at that, again, seeing the antigen-specific results, and being able to look at both of those and how those coincide with one another. So here again, we're seeing that with this priming signal with the HER2 positive CAR T cells that we are then seeing a significant elevation both in cell surface PD-1 of those immune cells as well as the PD-1 that is being secreted into the effluent. So now I'm going to pivot over, lastly, and being able to talk about looking at co-therapy. So in this case, we were using IL-2. So this is a very common co-therapy that is utilized with HER2 positive CAR T cells. And so with this, what
I
we
just wanted to give you like a 1 to 2 minute warning for timing wise.
Perfect. Thank you so much. And so with this, wanting to be able to enhance that CAR T cell performance, looking at IL-2 with those co-therapies, being able to see that we have an increase with IL-2 as a result comparatively to the killing capacity for based upon the quantification of total cast-based objects on the epithelial cells overall. And that this is, of course, can be looked at again in comparison with perforin, granzyme B, and degranulation, as well as increased cytokine release and looking at interferon gamma. So with that, in summary, I would just kind of say that CAR T cells are validated and be able to have a validated and flexible CAR T workflow that can be applied to a wide array of different solid tumor applications and models. But there's also then a lot of room for growth and being able to further possibilities for this application. I'd like to thank you so much for your time, and I will hand it back to Lauren.
I'm Tudor from Netri, a small company in Lyon, in France. And I will show you today how to profile PK profile in a microfluidic platform. So the main anti-cancer therapies currently rely on either biochemical methods or physical methods. And biochemical methods can include chemotherapy, hormone or immune therapy, stem cell transplant, or targeted therapy, including diverse type of nanotech. But also new therapies, which are being currently introduced, can suppose antibody-drug conjugates, tumor-infiltrating lymphocytes, and also drugs like PARP inhibitors, ATM and ATR inhibitors. So in order to evaluate the efficacy and adverse effects, anti-cancer therapies are all the time depending on administration, distribution, metabolism, and excretion. So in the whole process of drug discovery, from basic research to clinical trial, it is an important fact that we focus on defining a drug PK profile. Understanding and mimicking PK profile for anti-cancer drugs is critical for drug approval and also for patient output. So there is a constant urge to define and understand PK profiles. So these PK profiles can be either simulated in silico by in vitro in vivo extrapolation or explored in animal models, but this is also raising critical ethical issues and also interspecies PK variability. So some of the organ-on-chip developers started to explore also ADME effects in vitro. An anti-cancer drug PK profile can be simulated either in silico and then assessed in vitro, but only sequentially and using multiple bolus concentrations corresponding to different time points. So the optimal results in vitro are further assessed in vivo on animal models, and their results enter early phase clinical trials. And this is where the most failures are being observed. So what we are proposing is an in vitro approach to mimic any PK profile and therapeutical schedule for anti-cancer therapies. And our method can correlate data from in silico, in vitro, and in vivo and clinical trials. Using our devices supported by an automated system like Biomax 7, we can mimic any type of PK profiles on one or multiple drugs following customized therapeutical schedules. These are the platforms we are currently using, Netri organ-on-chip platforms called neobentos, which are used either for 2D cultures on the left side, like dual-link shift, and TRIA-link, and also platforms like duplex and duplex link, which are meant to work with 3D models and organoids. Thus, instead of using a classical bolus dose, we are mimicking PK profiles, and we are using a sequence of addition and removal of specific drug concentrations corresponding to specific points on the PK curve, as you can see. And also, we are able to program the time intervals of administrating this concentrations. These intervals can be set from 1 to 12 hours, but also trying to avoid excessive mechanical stress. At the same time, we can set customizable treatment schedules that may last for various time intervals from 7 to 21 days or more, but also depending on cellular models. Also, we are able to set monotherapies or combination therapies which are being spaced by washout intervals. And the open or semi-open architecture of our platforms allow time point sample collection for dynamic and endpoint assays. So as an example from a previous life in publication, which is using an in vitro colorectal cancer 3D model that was treated by irinotecan metabolite SN38 in monotherapy and in combination with an ATM inhibitor, I have compared the treatment efficacy according to treatment schedule and in vitro treatment setup. So literally, I was comparing the status bolus efficacy versus microfluidic PK setup. And the results, as you can explore in the paper with the link I have provided here, you can notice that the PK platform, the microfluidic setup, had displayed advantages over the static setup, and also the results were very, very close to those obtained in animal model, and also avoiding static setup over prediction for drug efficacy. Also, the proposed workflow is very straightforward. The PK is automatically generated with high accuracy, which was confirmed by mass spec analysis. The PK can be automatically customized, but obviously based on the in silico, in vivo, or clinical data using the automated platform. The same platform performs automated mixture for compounds if you have a combination therapy. These combinations, these mixtures are being delivered to the tumor-on-chip model also in an automated way. And also, these platforms are working with 2D or 3D tumor models. So, as a conclusion, our platform and associated protocol offers technical advantages as pumpless, mixerless, autonomous, and modular. It is able to deliver therapies according to customized PK profile and therapeutical schedules, including washouts. And also, it is working for monotherapies and combination of drugs and methods. And the advantages over animal models are that it is capable to explore single drug therapies, but also combination therapies that will become extremely costly on animal models. For example, if we choose the therapeutical schedule to include 3 drugs, this will require monotherapy, then combinations of paired drugs, and finally triple combinations, which is extremely costly, and it will use a massive amount of cells to develop the tumor model in animals. And also, animal control groups do not always survive for long-term experiments, contrary to 3D patient-derived organoids that we can use in our system. So literally, I have displayed a method that can be useful to explore efficacy and also adverse effects using a PK profile for anti-cancer therapies. So literally, if you're interested in more details, we will be very happy to let you know if you contact us. And thank you very much for your time. And I hope I have recovered the time gap.
Thank you so much, Tudor. That was a great presentation. We do have one question so far in the Q&A. And if anyone else has questions for Tudor, feel free to put them in the Q&A box now. So the question for you is, how does the in vitro setup take into account in vivo drug clearance?
That's a very interesting question. The drug clearance will require, obviously, some extrapolation and usage of additional microfluidic setup. For now, we are just exploring the efficacy. And using the washout steps, we can understand how things are working, but literally we cannot explore clearance without having additional organ on chips.
Great.
So we are likely to explore this using additional modules, but for now we are looking for exploring efficacy according to the PK profile. Yeah.
Great. There is another question. What is the type of sample being used in the examples you're showing?
So in the example I showed, we have used the colorectal model, spheroids derived from a cell line at that time, but I have also personally used colorectal patient-derived organoids.
Great. Thanks so much.
For Lauren for organizing this webinar, and it's a pleasure to present our solutions to use a scalable organ chip system for oncology applications. So just a few words about myself. So I'm the Senior Director of Advanced Research here at Xellar. Before I dive into the science, I'd like to just give you a few words about us. So Xellar is a small company. We started in Q4 2021 in Boston. Right now we have our headquarters and also a global business hub, as well as R&D center in Boston. We also have last year also set up a site in Asia, which is our site for manufacturing, Asian data center, and also a clinical lab. So what we do here is, we don't call us just an organ chip company. So we try to combine 4 different technology platforms to really, to better enable disease modeling, efficacy testing, safety assessment, and also personal medicine. So we start with our patented microfluidics devices, and with different designs and scalabilities. Using those devices, we then create different organ models. So from our most model are focused on the cancer, and which will be the topic of today's webinar. And also we have some efficacy, some toxicity models as well. Among all the readouts, we really excel using imaging as the main readout, being that it's a much cheaper and also high content readouts compared to other traditional readouts. And to better leverage all this data from the imaging as chemical information as well as omics information, we utilize state-of-art computational tools and AI tools to gain better insights into the biology. As the other speakers have alluded to already, there's an urgent need for better cancer model, not only for disease modeling, but also in drug development and personalized medicine. To give you a few, just highlight a few needs. So for there's or increased understanding of the complexity of the tumor immune microenvironment, and that is very hard to mimic using traditional 2D models or even animal models, where oftentimes they are immune deficient. For drug development, as we know, historically the translational gap from preclinical to clinical is pretty low, and this is especially true for oncology. You can see that the average success rate for oncology drug is less than 5%. So we are defending more predict models with higher predictive validity to accelerate the drug development with greater efficiency. And finally, for the patients, we know that with the advance of targeted therapy in the last decade, there has been great progress, but still less than 5% of the patients actually benefit from targeted therapy. So we need better ways to select therapeutic options for individual patients, and ideally with quicker turnaround. As you know, generally we agree that the model accuracy to mimic human disease or drug responses to drug scale with the model complexity from simple 2D models to organoids to non-human primates. But there are some deviation from this model due to many interspecies differences, as especially true for cancer. So what we do here at Xellar is we try to create models that retain the complexity or the human mimic as much as possible, and without losing too much on the scalability. To do this, we have created different organ chip platforms with, as you see here, from here, different designs. Let me go to my laser point with different designs, different throughput to meet the needs of different applications for different disease and also for different user application, either a model development or high-throughput screening. Our device have a few as many advantages, but I just want to highlight a few of them. For example, they are 100% thermoplastic, so it has it's an injection molded, so it's really amenable for mass production. And also, it's we primarily develop imaging-based readout, being that it's a much cheaper assay and also provides much more information than just a single readout. And then among all the different organ chip devices we have created, this is one of them called AlveoliX 32. So in a standard 96 plate format, it has 4 independent devices. Within its devices called AlveoliX 8. So this is a 8 chips device. It has 2 individual channels. One is called gel channel, which can be used to see like cells embedded in the 3D matrigel or other sort of hydrogels. Spheroids or organelles can also be mixed with the gel and seated in this channel. And then the adjacent channel is called a perfusion channel. So this channel can be used to perfuse culture medium, either using gravity-based flow or using a peristaltic pump. This channel can also be used to seed endothelial cells and provide a better, a more complex environment to mimic the tumor microenvironment, as well as the flow of different types of immune cells. This is a typical experiment flow of our consortium model. So starting with AlveoliX 32 devices. So we have successfully worked on with the cell lines, organoids, immune cells, and stromal cells to first create this model, and then we can test the efficacy of different types of therapy, including chemotherapy, targeted therapy, immunotherapy, and also cell therapy to the device, and then use different readouts to measure the drug efficacy. So including blood field images, live diet, apoptosis, and some other functional staining. We can also use traditional ATP-based measurements to quantify the number of cells that's caused by the therapeutic candidates. So just give you an example here, we used pancreatic cancer cell lines, BXPC3, in this model and treated with chemotherapy. We can see the dose-dependent increase of cell death. And this can be quantified using the cell title glow or the ATP readout or imaging readout by measuring the viable error on the chip. So as you can see that there's much less variation using the imaging-based readouts than the gold standard cell title glow assay. But overall, they have pretty good correlations. And the conclusion is that imaging, of course, is cheaper, faster, and potentially more accurate than the ATP assay. So using this feasibility model we have created, then we use some primary patients-derived cancer model to perform limited POC studies to test the ability of our model to predict patient response. So in this case, we used the tissues from a patient with squamous cell cancer, culture them on chip for 6 days, and then treat them with cancer drugs for 3 to 6 days. So here we have created a deep learning-based algorithms to automatically segment the cells of interest and also to classify the live cells and dead cells and use that information to quantify the drug response. So just want to highlight, we did this comparison between traditional 2D culture and on-chip culture. And notably, you can see that one of the drug called Microphysiological is it's not recommended for this particular cancer, squamous cell lung cancer. However, it shows pretty profound effect on 2D culture, whereas it has no effect on chip, either treated on chip for 3 days or 6 days. So this is indicating that our model has a ability to predict a patient response. Right now, we are doing this kind of study in a much larger clinical, using much larger cohort of patient samples. So this just give you one example. We collected, in this case, tissue from colon cancer patients. We can culture this, of course, using traditional matrix overlay organelle culture, but also we can culture this on chip. You can see the growth of the cancer cells over time, and then we can do all kind of or use different approaches to characterize our model shown here, including the comparing the biomarker expression on chip comparing to that of from the original tissue. We can also use imaging, including blood flow images, to examine the drug effect on the individual object, meaning the individual cancer organoids, and also use the lab data staining and imaging analysis to provide a better quantification of the drug effect. So we can do this. So this is just for with monoculture. More importantly, given that our devices features like a barrier-free design, so that allows direct cell-cell contact as well as cell migration from one compartment to another. So in this case, we cultured as a proof of concept. We cultured a human lung cancer cell line FF19 cell labeled with a GFP tag, and then human vascular endothelial cells on the adjacent channel. You can see the migration and invasion of the cancer cells from the gel channel into the perfusion channel, and we've done studies to show that this led to a decrease of the barrier functions and also leads to the recruitment of immune cells. As an example, on the right side of the graph, we cultured, in this case, XVIVO. So cancer cells from mice, and then we use M2, in this case, M2 macrophage, co-culture them both in the gel channel and show that they promote the growth of each other. In this case, the LLC1 is a cold tumor, so in the presence of M2 macrophage induces proliferation. After we introduce CD8 positive T cells to the perfusion channel, we can use the live cell imaging to visualize the migration of the, in this case, green labeled T cells from the perfusion channel into the gel channel, and that leads to the killing of the red labeled cancer cells at the 48 hours after the cell were introduced into the channel. So that was how we create this model. So what we, as I mentioned earlier, we really like to scale up this from feasibility to much larger capability to a much larger screening. So how do we address this scalability issue? We have 3 different approaches. First is we have a larger format devices. So in addition to the AlveoliX 8/32 devices, we also have a AlveoliX 96. So it has a per channel, it has the similar design. So that meaning all the protocols we have developed from the small format devices can directly translate into the large format devices. And we feel that under dynamic conditions, the cell grow faster, and also the cancer cells tend to be more sensitive, bringing a lower IC50 when they are in that dynamic culture mode. Our devices follow SPS format, so we can automate the entire process from the cell seeding to drug dosing and to the endpoint lab data staining. We show that with the manual and automation, you get similar drug response curve. Finally, we from all these images we collected, and we have trained the AI algorithms that will allow us to do a so-called in silico staining, meaning that using the bright field images to predict the lab data staining. And by doing so, we show that we get a more sensitive measurement of the drug response. So with that, I'd like to end my presentation. If you have any questions, please feel free to reach out to me personally, or you can find us through LinkedIn or through our website. Thank you.
Thank you so much for your presentation, Haiking. There is so far one question in the Q&A, so if anyone else has questions, feel free to put them in now. The first question is, what is the size of spheroids that the chip can hold?
The largest one will be about 200 micron in diameter.
Thank you so much. It's wonderful to be able to speak with you today. And I'm going to be speaking on how we can use our simulated tumor microenvironments in our organ-on-a-chip models for drug safety delivery and efficacy studies. We all understand, that a tumor microenvironment is very complex, but it absolutely impacts drug delivery, safety, and also efficacy. And one of the reasons why is because the microenvironment oftentimes has a variety of different physiological parameters that have to be navigated when we're doing drug delivery. That includes oxygenation. So the distance that's from the solid tumor is from the blood vessel can lead to everything from a physoxia, environment to hypoxia and even a necrotic environment just based on the distance. And as that solid tumor grows, we also know that it is proactively modifying its own microenvironment, and that facilitates not only uptake for oxygen and glucose, but it changes the extracellular matrix and impacts the other types of cells that are localized in that environment, from fibroblasts to tumor-associated macrophages and endothelial cells. And as the tumor initiates its own blood supply and finally can enter within those microvascular vessels and then metastasize, we again know that the genetic and genomic changes that can facilitate that are also responsive to that microenvironment. In the end, modeling all of that on chip is very difficult. And so the ways that we can utilize that is through some unique architecture designs. So I'm the Director of Scientific Development here at SynVivo, which is an organ-on-a-chip platform that has different, aspects that and features that can help for visualization, quantitation, and examining these different components of the tumor life cycle and its microenvironment. So we have a side-by-side architecture with prefabricated porous barriers that allow us to lay out our tumors and our vasculature in a way that allows direct cell-to-cell contact and migration, as well as exchange of factors and also differing oxygenation and glucose environments that can impact the tumor growth itself. Within these microvascular designs, we have digitized scans of in vivo human and murine vascular beds that are then placed on our chips, which is a PDMS layer on a glass slide. Our microvascular beds are 100 microns wide by 100 microns tall, and our tissue beds themselves are also at differing widths and parameters, but also 100 microns tall. So when we're seeding cells into these organ-on-a-chips, we tend to use dissociated tumor cell products that are coming from biobanking, or we're using different cell lines, and we can resuspend all of these products into hydrogels and directly inject them into the tumor microenvironment. So we can do cell mixtures, or we can just use a solid tumor cell line. And with the variety of different architectures and designs that are available, it allows you to really tailor the type of microenvironment that you want to create. Whether it is a microvascular bed that sustains a single tissue area, or a more idealized design with a wider tissue zone shown here in red. We also have multi-chambered chips that allow for different perfusion profiles to really create a very dynamic tumor microenvironment that directly impacts activities such as metastasis and drug delivery. And we can have it where the different tissue areas are adjacent to one another, or multiple tissue beds that are separated by their own vascular beds, meaning that metastatic cells actually have to cross into the vasculature and become circulating to invade the secondary sites. So all of these features enable the characterization of these cells within a dynamic environment. So I want to describe just some different examples of how that looks on chip, and then what kind of readouts that we achieve when we are simulating these tumor microenvironments. In this case, we have a microvascular bed with 3 different tissue areas highlighted in red that are separated by a 100 micron wide microvasculature shown here. We are seeding the tumor cells in a hydrogel mixture in this first area labeled number 1. And we are comparing, in this case, 2 different breast cancer tumor cell lines. One that's a metastatic tumor cell line, MDA-MB231, or a non-metastatic line, MCF7. And both of these are GFP labeled, as you can see here. But you observe that the MDA-MB231 is very proactive in terms of metastasizing not only into the vasculature areas, but directly into these tissue zones. And whereas the MCF7 really stays kind of localized to that tumor area 1, that site 1, where the cells were initially seeded. If we utilize a fluorescent tracer and place that tracer through the vasculature to just observe how the impacts of permeability change with different tumor types, what we can observe is how a metastatic cell line really can kind of degrade the extracellular matrix and really promote permeability. So if we look here in the red bars, this is where the FTSE-dextran is introduced as a solid, and you see it's at full saturation for all the models. And then in the blue, you can see how much of that tracer actually flowed into the tumor area itself. And from our control, you can see very little pass through, but with the metastatic MDA-MB231 cell line, we see a really sharp increase in that fluorescence intensity, really indicative of the promotion of that metastatic cell line to breaking down the barrier integrity of that microvasculature. Whereas the MCF7 non-metastatic cell line is very similar to controls. And then we have a pro-inflammatory cytokine that we've introduced into the vasculature as a positive control for paracellular permeability and barrier integrity breakdown. And that's more similar to our metastatic line. So you can really see the impact of metastasis on vasodilation within the system. But in addition to that, the delivery of drugs across this microvascular bed also increases its complexity. So we have 2 different types of perfusion chips. One is a high perfusion chip, and we have a low perfusion chip shown here. And when we're talking about perfusion, we're really talking about the accessibility of the media to all sites of that initial tumor seeding area. So in red here, you can see zones where the actual distance between these tissue areas is quite low, and it's fed on either side by microvasculature. And so you see a high perfusion of nutrition and media in those areas. But there are zones that are spread a little bit further apart. The microvasculature can only sustain a smaller amount. And so you can see really kind of the drop off in terms of the amount and areas that the glucose and the pH can be regulated. In the lower perfusion chip, you'll notice that the red zones are much smaller, but these zones of blue really stand out. And that allows us to kind of generate zones of hypoxia and necrosis within that solid tumor, that 100 micron thick tumor. When we deliver drugs through the vasculature to then treat the tumor and observe its cytotoxic impacts, what we see is with the high perfusion chip, we can really see a decline in both a non-metastatic and a metastatic cell line because the perfusion of that drug is accessing more areas of the tumor. However, in the lower perfusion chip for doxorubicin treatment, we can see that there's an increase in the overall viable tumor area, indicating again that the drug is penetrating less throughout that tumor zone. But if you have a drug with a differing mechanism of action, like paclitaxel, you can observe some differing effects. For example, in the higher perfusion chip, you see a drop in the non-metastatic cell line in terms of overall viable tumor area that's slightly different from the metastatic cell line, the more aggressive one. But in the lower perfusion chips, again, the viable tumor area is more sustained because less of the drug penetrates through, but you can see some greater sensitivity in the metastatic cell line compared to the non-metastatic cell line based on the mechanism of action. So these different type of chips really can help you as you're trying to facilitate your drug delivery and drug efficacy studies. So from there too, these chips also allow us to look at circulating tumor microenvironments where we have circulating tumor cells. So once the metastatic cells have entered into the vasculature, we can observe their circulation through that microvasculature, especially if they're GFP tagged. But in addition to that, we can also introduce a circulating immune component on chip. This includes fluorescently labeling monocytes and flowing them in and allowing them to enter into across the porous barrier and enter into the tumor zones where they can start their killing effect. And you can see that co-localization between tumor cells labeled in green and the THP1 cells in red, and it shows up as yellow there. So our platform is highly visual, but it allows you to look at both circulating tumor cells as well as immune components. When it comes, though, to drug delivery, a lot of times we want to evaluate whether or not our drugs are successful at entering into the target zones and areas and successful at killing without having a lot of off-target effects. And we've utilized enhanced permeation retention, EPR effect modeling to try to determine how that occurs within our microvascular beds. As you can see here, our MDA-MB231 cells really reduce junctional VE cadherin expression, which is common based on the metastatic nature and the collagenases and other MMPs that this cell line will secrete. And so that really leads to an increase in permeabilities we've seen before, which is much less than our MCF7, which doesn't impact the tight junction proteins quite as much. So you see a much more intact vascular barrier. However, when we're looking at delivery of liposomes through a passive targeting mechanism, the tumor microenvironment absolutely influences that enhanced extravasation. So because the MDA-MB231s are already actively breaking down the endothelial barrier, you see a significant increase in the amount of liposomes that are able to reach that solid tumor through that vascular barrier, which is much less than an MCF7, non-invasive, and so the vascular barrier is a little bit more intact. However, this can also be looked for targeting, where the liposomes are carrying specific antibodies that are allowed to bind to targets such as E-selectin. Because the vasculature is so inflamed, the E-selectin will be upregulated in a metastatic cancer model. And so you can see that the targeting and the infiltration of the liposome significantly increases when their targeting mechanisms are available. So in cases for nanoparticle deliveries as well, we have demonstrated that our nanoparticles can be studied on chip in a non-autologous system and with whole blood. I won't go into as many of the details for this particular study, but you can combine and introduce whole blood onto the chips and study the impacts of nanoparticles and look at their binding to immune cells under static and flow conditions. Our models are kept under dynamic fluid flow, and so that allows us to monitor these kinds of interactions and determine if immune cells are acting as a sink for nanoparticles instead of the nanoparticles reaching their desired target. For drug safety as well, nanoparticles or nanocrystals can be utilized and flowed through the vasculature to mimic, a chemotherapeutic delivery. In this case, we have unmodified nanocrystals that have a quite important and very toxic side effect, as you can see here, both to the tumor cells and also to the endothelial cells. They're very cytotoxic. However, if you modify these nanocrystals with folic acid receptors, you'll notice that the toxicity onto the endothelium significantly drops, but it does still retain some tumor cytotoxicity. So you can utilize this SynTumor platform for screening modified nanoparticles, again, for delivery as well as safety and efficacy. We've also done some work with the National Research Council in Canada on CAR T cells flowing through in an iPSC-derived brain endothelial cell model. We see the extravasation of these CAR T cells into a glioblastoma cell line that's effective at leading for killing. We can also look and see if there's any damage to the endothelium, both before and after CAR T administration, to prevent some off-target effects, again, that lead to swelling on the brain. And finally, we can look for drug efficacy at tumor invasion of secondary sites, and this can be done over a longer period of time. So we have MDA-MB231 that's incubated here, and we can look over a course of 26 days to really study its invasion of a secondary site and progression out through that. And again, with these particular models, you can also introduce chemotherapeutic drugs within the vasculature to see if this effect is halted. So overall, our SynTumor platform is very versatile. It's a very visual platform, but can also be used for quantitative assessments, and we can collect effluents and cells from these systems to really evaluate solid tumor targeted therapies. Our vascular geometry mimics the in vivo microvascular beds and simulates these complex tumor microenvironments that allows us to look for drug delivery, safety, and efficacy that can be modeled on chip and using some intravenous drug delivery methods that mimic the chemotherapeutic protocols. And overall, it's very critical for us to be able to mimic these tumor microenvironments so that our anti-cancer therapeutics do lead to fewer off-target effects. Thank you very much.
Thanks so much, Deborah. That was great. We do have a few questions for you in the Q&A, so I'll just read them out. The first is, how do you monitor permeability?
Yes, so you can monitor permeability in two ways. One is through using reagents that monitor paracellular permeability, so you can use fluorescent tracers like dextrans or others if you're wanting to have a fluorescent readout. We also have a tier on chip, so if you're wanting to do a very non-invasive way of monitoring changes in barrier integrity, you can utilize the transendothelial resistance and monitor that over time as well.
Great, thank you. A second question is, you mentioned CAFs, TAMs in the intro. Do immune components of the TME have representation in any of your chips?
Yes, so the way that you can integrate innate immune components as well as other types of immune cells that might be resident within a tissue is we will mix them in different ratios with our tumor cell products. For some of dissociated tumor cell lines or products that you can get from different biobanks, they often come with the immune component already as a part of it because it was a part of the resident patient tissue. So if you take those dissociated immune products, especially like for lung cancer, you can resuspend them within a hydrogel and inject those directly into our devices. That way you have a very complex combination of both tumor cells, normal epithelial cells, and immune components too. And so you can put the immune cells through the circulation, or you can also embed them within the tumor channel so there's direct cell-to-cell contact.
Great. Thank you so much, Deborah. I think another question just came into the chat, so we have time for one more. How do you adjust for frequent culture media change and the small tissue samples?
Yes, so for the frequent media changes, our system is connected up to syringe pumps so that the maintaining of the system can be ongoing over several days. Within the tumor zones and tumor areas especially, it's a very hydrated area. So again, the hydrogel matrix keeps that area very hydrated. And then you get sustenance coming from the vasculature as well. So the tissues are sustained by the vasculature as it would be in vivo.
All right, so those of you who aren't familiar with Hesperos, it was started by Mike Schuler and I about 9 years ago. Mike Schuler is considered the founder of the body in a chip field. Both of us have been working in the area of MPS for about 30 years. We have previously won the Lush Prize for animal testing. We've used a lot of SBIR grants to get the company going. We work with folks all over the world. We got a 14,000 square foot facility here in Orlando, Florida, which we're expanding as of this moment. We have about 50 people at the company right at the moment. We're a CRO. We do not sell our systems. I won't show this during the presentation, but we have a wide variety of different CNS and PNS models. And one of our peripheral nervous system models was the first microphysiological system where only our efficacy data enabled a successful IND that led to the authorization of a phase 2 clinical trial for a rare disease. Began recruiting patients in April 2021. That was actually a successful phase 2. It's now in a 2-arm phase 3. Again, this was done in collaboration with Sanofi. There's a publication out there on this. Happy to talk to it. But basically showing that we're getting regulatory acceptance of our models and also potentially clinical predictivity. So what are we doing differently than a lot of folks? I've been working in this area, what I call clinically relevant functional readouts, for about 30 years. So what does that mean? You go to a doctor's office, he doesn't immediately stick a needle in your arm to look at, take out fluids, look at biomarkers, right? Looks how you're walking, how you're talking. He listens to your heart, he checks your reflexes, he might ask you about elimination. What he's really looking at is, are your muscles contracting okay? Electroactivity, your neurons and cardiac cells functioning? Is your neuromuscular junction also working? Is your GI tract and kidney activity working well? So if the barrier integrity is there. And the reason why I'm going through this in terms of cancer is what we really focus in is the off-target toxicity for cancer cells, but also drug-drug combinations for chemotherapeutics. And we have the systems to be able to do that in the same model. And what we can do, we can use these non-invasive clinical readouts for things like cardiac force, where we can put thin layers of silicon, put cardiac cells on it, so when it contracts, it deflects the laser, we can get force. We can pattern cardiomyocytes, get conduction velocity, more importantly, QT interval. That is a major target for looking at off-target tox for chemotherapeutics. We can look at skeletal muscle the same way. Again, we have a wide variety of different neuronal systems, CNS based upon long-term potentiation, conduction velocity, neuromuscular junction models, which we can also integrate. We can still look at biomarkers, say, for the liver, but we don't have to. This allows us to look non-invasively, but more importantly, chronic monitoring these systems. We've actually published at L'Oreal taking these systems out for 28 days. But we're doing this all without cell death, okay? We can still monitor cell death, but we're really trying to see what are those clinical readouts you'd really want to be to know, okay, that are making the organism not functioning properly that might eventually lead to cell death, but currently aren't doing that. So how does that work? What we do, we can culture out the cells, okay, on their own individual chips in their own media, and then time it so they're all ready at the same time to put into a base platform. We then put a gasket on top where it defines a fluidic channel. So we can actually then flow directly. We're not pipetting from one chamber into another chamber. We actually have recirculating media in these systems. All this is in serum-free media. We can get multiple organs in serum-free media recirculating out for long periods of time. And then we can then put a clear top on it so we can still look at all the imaging technology that's been developed. So what we can do, we can take out, say, this chip and put in a pancreas chip, and again, because it's so reconfigurable and flexible, we can, all the different cancer models you've seen, we potentially could integrate into our system at the same time, be able to monitoring the off-target tox for things like brain or kidney or liver or cardiac-based systems. How do we do that? Mike Schuler had basically taken the equations used for predicting phase 1 dosing, modified those, and this is the first design multi-organ system that he published back in the early 2000s. This is one of our more advanced systems I won't get into, besides just saying that we have used this for opioid overdose and looking at naloxone recovery, but at the same time monitoring off-target tox. And again, these are all the references that be able to lead to this. But I really want to highlight the fact is all this is done in a pumpless platform, which means we get rid of all the tubing, we get rid of all the dead volume, all the pumps, and we use gravity to pump the system. It also makes it a low volume system. So I have a competent liver system. I can look at the effect of both parent and metabolite in the system at the same time. Just to show you, build up the different tissues we can put into it. We've been able to take hepatic cells and do 2D modeling as well as 3D modeling with these systems. We don't have to use stellate and Kupffer cells. We add them in selectively as variables, but this is just with hepatic cells. We've done a wide variety of SIPs, but in this particular paper, we did 1A1, showing nice basal activity, and 3A4, and also induced activity. This is published in Biotechnology Progress. And we can then combine that with the cardiac system. And this was a project we did with AstraZeneca. AstraZeneca had a molecule flown through preclinical, but as soon as they put it into an animal, they saw a huge QT effect. And so they said, okay, let's validate it first with terfenadine, fexofenadine. Terfenadine is an antihistamine that was pulled off the market because it caused a large QT effect, whereas its metabolite, which is fexofenadine, currently marketed as Allegra, does not. So you see in the heart-only system, we see a large QT effect. soon as we put it in the liver, we see it shift far to the right. Conduction velocity in the cardiac system is not affected if the liver is present, but you see an IC50 of 13 micromolar if it's a heart only. But again, the viability is not being affected. We're doing all this without cell death. So I want to show you, those are two of the components I'm going to talk about in some of the later systems, but also I included this paper to show that we can also grid up the system. We can model it in terms of flow rates, in terms of shear stress. We can monitor for compound absorption. It doesn't matter what the plastic is. You get some absorption of compounds to all plastic. If you try to minimize hydrophobic, you will get some hydrophilic and vice versa. In this case, terfenadine is a highly absorbing compound. Okay, in our platform, fexofenadine, the metabolite is not. So what we can do, we can then monitor for mixing in the system. We can look at a loss due to the absorption, but also loss due to metabolism, get the area under the curve. 95% of the systems that are out there is as dosed. We have a full analytical capability for looking at HPLC-MS to be able to look at real concentrations, get the area under the curve. And by being able to create the area under the curve, we create these pharmacokinetic pharmacodynamic relationships where we can monitor using controls for the amount of absorption in the system. We can monitor the amount of metabolism to be able to get the pharmacokinetics for each of those. We can then use the functional readouts, again, which are mimicking what you'd see clinically, in this case for cardiac, to be able to get the pharmacodynamics. In this particular case, terfenadine is actually approaching the hERG channel from inside the cell. So we can actually look at the pharmacodynamics of the what's going on inside the cell. We were able to use this data for our PK/PD for the in vitro system to be able to then combine that with in vivo PK data to be able to predict the pharmacodynamics for guinea pig, dog, and non-human primate. You see very good correlation with this data, and this was published in Nature Scientific Reports. So we can take these systems all the way through to be able to start doing predictions of dosing in for clinical trials. Now, for getting to the cancer aspects, what Roche did is they said, well, okay, that's a very nice toxicity model. Can you combine it with an efficacy model for a multi-drug resistant cancer and a non-multi-drug resistant cancer, at the same time monitoring off-target tox? And so they picked tamoxifen. Tamoxifen by itself is not a very good chemotherapeutic. However, its metabolite is. So you see in the non-multi-drug resistant cancer, you see when you add it in the liver, you see a decrease in proliferation. With the metabolite, you see a decrease in proliferation. But for the multi-drug resistant cancer, you can basically see here you don't get any effect with tamoxifen, with the liver present, or in the metabolite. When we add in a second drug, verapamil, which blocks the PGP channel, and the PGP channel is basically pumping out the chemotherapeutic as fast as it goes in, you do see a decrease in proliferation. You don't see an additional decrease in proliferation, so you know verapamil is actually blocking the PGP channel and not acting as a chemotherapeutic. Now, the interesting thing, and this is the really take-home message for the entire presentation, is that we then could monitor the off-target tox in the same system for both parent and metabolite, okay? And what you see here with tamoxifen by itself, you see a decrease in conduction velocity, but recovers, decrease in force, but recovers, small decrease in beat frequency, but recovers. But for this drug-drug combination, and this is really what we're working on mostly with these models, is the drug-drug combinations, because people have some idea of how the efficacy combines for a lot of chemotherapeutics. In many cases, they really have no idea how the off-target tox combines. And in many cases, they're having to go back through for drug-drug combinations for into some animal experiments to actually prove that those toxicity. And again, this is a demonstration of that, in that this drug-drug combination, the conduction velocity goes down but does not recover. Force goes down but recovers slightly. Now, beat frequency goes down, but recovers to some degree. Now, if you're only monitoring beat frequency, you would say that, okay, there's not a really bad off-target toxicity for cardiac for this, but in actuality, if you're measuring the really more advanced readouts, you will see that's the case. And again, at the same time, we're not getting any cardiac viability changes in this system. At the same time, again, no liver toxicity with this model. This was published in Science Translational Medicine with Roche. It was also featured in Francis Collins' blog. So this is the crux of what I'm trying to present, which is we can look at your drug-drug combinations, monitor efficacy for whatever cancer combination you would like to have, but at the same time monitoring the off-target toxicity, okay, to give you some idea. And to take that a step further, I'm going to show some results where, again, we have taken these systems with L'Oreal to do selective drug responses, published back in 2016 for a 4-organ system. We had also taken with L'Oreal again in advanced functional materials, taken this out for 28 days, showing function in this model. And what we did is we used that 4-organ model with Vanna Pharmaceuticals. The FDA had asked them to do a 9-month Beagle study for studying toxicity. Beagle is normally used for cardiotox, but can be used for others as well. And so Van approached that, said if they wanted to combine their 3-month Beagle study with a 2-week study using our 4-organ system with their compound with proper controls, okay? And that data combined with a 3-month Beagle study allowed that to get into review without having to do the 9-month Beagle study. So we're hoping to try to get these systems, okay, where you could use one of these multi-organ systems for both the efficacy and off-target tox to potentially avoid the animal, going back into the animal studies to be able to look at the toxicity for the drug-drug combinations. So again, just showing that we can get in the multi-organ system, good morphology in these models. Okay, you see again that the patterning, you see the neuronal systems, very good communication between the electrodes, hepatic cells. And what we did is we did 4-organ system, and then they get their compound plus 2 different controls for each organ system and monitored that out for 14 days. So their compound, tradipitant, again,
James,
didn't show
I just
much.
wanted to give you a 1 to 2 minute warning for time.
Okay. We don't see much with their molecule in terms of liver toxicity. We do see for troponin, we see a large effect, but then it goes down. Again, we see for the 2 control compounds, we're getting toxicity in terms of the force readout, but not for their compound. Okay, beat frequency, again, going down for the 2 controls. For cardiac, for QT interval, again, see that going down, but see a spread of that for the 2 control compounds, but not with theirs. Same thing with kidney. You see one compound is very toxic, one which is partially toxic in the system. And then you can see for neuronal activity, we get very good correlation. And it is actually a very interesting dose response curve depending upon, this was all done with a low metabolizing, mid-metabolizing or high metabolizing liver. So we really could see a population dynamics without having to go to a large number of different systems. We could actually then look and see in terms of the level of CYP activity in the model. Lastly, we've also can put a recirculating monocyte culture in this to be able to look at the innate immune system. This was published with Roche in Advanced Science. We're able to show the development of either M2 or the M1 phenotype in the model system. So thank you much for your attention. Again, our business model is we will custom build what you want, and we'll also tell you we can't do it. But really focusing on you've seen wonderful efficacy models. We're really focusing on the off-target tox, especially for drug-drug combinations with these model systems. Thank you.
Thank you for the opportunity to present AlveoliX technology during this workshop. Today is my real pleasure to show you how our long-on-chip model can be used for cancer therapy safety study. As you may know, AlveoliX is a Swiss biotech company specialized in the development of organ-on-chip technology. We have notably developed a system called Barrier-on-Chip that you have here. It integrates different engineering and biological parameters that allow us to mimic the dynamic microenvironment of several organs. So typically, we can reproduce the breathing motion on the lung, we can reproduce the peristaltic movement of the gut, the muscle contraction and relaxation of the bladder, and so on. So this technology is composed of 4 different key components. We have the AX12, which is our consumable. It's where we are culturing our cells. I will detail that a little bit after. We have the exchanger that allows us to exchange medium inside our chip. The actuator, which make possible to recreate the mechanical stretch. And finally, we have the dock station that makes the interface between the pneumatic component and our consumable. So the chip itself, the AX12 chip, I've been engineering to follow a 96 wet plate format, and it contains 12 independent units. What I call a unit, it has the following. So it's composed of an inlet, a cell compartment, and an outlet that are all connected by microfluidic channel. So the inlet and outlet well enable to change medium inside the basolateral chamber. And on the middle, we have a thin and porous elastic membrane that serves as support for cell attachment. So on it, you can culture different types of cells and adapt it in function of your need. So you can opt for mono, co, multiple culture. Here, for example, we have an air-blood barrier with immune cells. To complete the microphysiological environment, our technology allows the introduction of mechanical stretch. So for that, at the bottom of the plate, we have a cavity, which is here in gray, and the membrane can be pulled down and up, and this deflection is transmitted to the AX membrane where the cells are. So, for example, you have here the 3D deformation of primary alveolar epithelial cells. So we can really reproduce the CD cyclic stretch which occur on the lung. One interesting thing to know is you can actually tune several properties to recreate the mechanical stimuli which occur on different organs. So you can opt for a uni or bidirectional deflection. You can adjust the frequency, the amplitude, introduce dormant period to mimic the circadian rhythm. So you can really tune it in function of your needs. Today, I will really focus on the lung. But before I'm really talking about the application on oncology field, I would like to mention that our chip is highly compatible with currently established readouts. So we can measure barrier function, protein cytokine release, we can measure gene expression, we can do live fluorescent imaging, we can also do most of the innovative omic readouts such as transcriptomic or proteomic. So it's really well adapted for several applications. So let's see now how we can use this technology for cancer immunotherapy safety study. So as you may know, cancer immunotherapy includes all kinds of therapy that harness the immune system to fight cancer. So it includes different approach and medication ranging from monoclonal antibody to small cytokine used to regulate the immune response and so on. And despite the really huge potential, immunotherapy presents a high risk of immune-related adverse events as well as the high risk of off-tumor toxicity. So the safety assessment here, it's really crucial. And for that, we can use predictive preclinical models that are human-based, since the human system of animals differ from the one that we have for human. So the first case study I would like to share with you today is on the safety profiling of T-cells bispecific antibody. So such antibody are engineered to recognize both T-cells and a target. So basically it works by bringing close the T-cells to the target cells, and this creates a functional immune synapse that leads to target cells killing. And in our case, the target is a folate receptor 1, which is a molecule which is overexpressed in certain type of cancer. but unfortunately, it's also expressed in healthy tissues such as on lung, on kidney, and this is the reason why TNCH TCB present a high risk of on-target off-tumor toxicity. So when this compound, this 4L1 TCB, was tested in mice, it showed a high efficacy for tumor killing. But it also showed severe lung toxicity in monkey with immune cell infiltration in the lung and respiratory inflammation. So that's why at the end, the clinical development was stopped for this molecule. So we wanted to know if with our technology, with our chip, we will be able to recapitulate this toxicity on human cells. And for this, we have cultured patient-derived alveolar epithelial cells on the membrane with immune cells. And we treated our cellular model with 4L1 TCB and also with a non-functional TCB variant as a negative control. The first thing that we checked was the target detection. So we could see that after culture on the chip, the cells were still expressing 4L1, and we confirm it via immunofluorescence and also via gene expression. We even observed an upregulation on the chip, which was actually expected since we isolate epithelial cells for culturing on the chip, and it's where the target is mainly expressed. So how the target was well expressed, we perform the treatment on the chip, and we measure several functional readouts, notably the alveolar barrier function. So we could observe here by measuring the TER that we get a drop in barrier tightness of more than 70% with TCB at 0.1 microgram per milliliter. And on the right, we can see that this concentration leads to the release of several inflammatory cytokines, so notably interferon gamma, TNF-alpha, or granzyme B. We also investigate cytotoxicity via monitoring Caspase-37 by live imaging, and we found that TCB increased the apoptosis signal compared to negative control. And we also checked for LDL release, and we found that the high dose of TCB caused membrane damage. We also checked for the recruitment and activation of immune cells, and we could observe that in presence of TCB, immune cells were specifically recruited to the epithelium. So here, the number of immune cells was significantly higher on treated sample. So we also analyzed actually by flow cytometry these immune cells from the chip, and we could find that both CD4 and CD8 were activated in presence of TCB, and we could even detect some degranulating CD8. So we show here that with this model, we can predict toxicity in the same way that what have been found in monkey study. And so that means that this technology could be used for the evaluation of potential new variants to help in preclinical decision making. Just to give you an idea, this data was already used in 5 IND submissions. The second case study that I would like to share today, it's about drug-induced vascular leak syndrome that happened, for example, with interleukin-2 immunotherapy. So interleukin-2 is used at high dose as immunotherapy for metastatic melanoma and renal carcinoma. And despite the efficacy in targeting the cancer metastases, 1/3 of patients treated with IL-2 develop a vascular leak syndrome. And this can lead to severe lung condition, which needs sometimes the incubation for 5 to 12% of patients. So we wanted to know if we could reproduce this clinical observation in our chip, and especially this patient variability, to check if we can predict if the patient will develop a vascular leak syndrome or not. So for that, we culture human alveolar primary cells from 12 different donors with endothelial and immune cells. and we treat it with IL-2 at physiological relevant dose, which means here with the dose that we found in the serum of patient that gets the treatment injection. So the first thing that we check was the inflammation response. So we ran a panel of inflammatory cytokine, and we find that some of them was upregulated upon AL2 treatment, with a similar trend between epithelial and endothelial compartment. And among this, I would like to highlight two clinical markers for the IL-2-induced vascular leak, which are the 6-CL10 and interferon gamma, which are known to be upregulated upon EL2 treatment in patients. And actually, we was able to see that also in the chip. When we check for the barrier function, we find quite some variability among the 12 donors tested. And it was actually something that it was expected, as in clinic, some patients developed the syndrome while some other was not. So we wonder if we can find a pattern to group these different donors. So for that, we hoped for a very basic approach based on TR, as it's a functional readout for leakage. And what we did is we grouped them with a different donor in function of drop of TR at the end of treatment. So in this way, we created two groups of donors, one with a patient that have a high risk to develop a vascular leak syndrome, and the other group, which was safe regarding VLS. Then we have a look to different readouts, starting by barrier function with these two groups. So we thought that the one we get the drop in TR was well correlated with increased permeability in the risk group, while we didn't detect significant difference on the safe group, what with small or even with larger molecule. In the same way, regarding cytotoxicity, we observe on the epithelium a slight increase in the VLS risk donor, but not on the safe group. So we also could note that actually there was no significant difference in cytotoxicity for the endothelial cells, only for the epithelium. So we show with this model that we get an inflammatory response in all donors, while the barrier impairment and cytotoxicity were found only in subgroup of donor. And even with this basic approach, we can group the donor that we have. So with the second case study, I show you that with our technology and with our model, we were able to capture the donor viability, similarly to what we could observe in the clinic. So to conclude this talk, I'll show you today how our technology have been used for safety study and the impact it could have on preclinical decision-making, END submission, in capturing donor viability. But I also would like to mention that some of our partners are using our technology to develop efficacy model, notably to study the effects of drug on bladder cancer on chip. So this is also possible with our technology. So with that, I would like to thank you for your attention. And if you have any research which is matching with what we are doing, don't hesitate to contact me.
So thank you everyone for staying here online. Thanks as well with Rare Collaborative for this great opportunity to talk. I would like to present ourselves. We're a bit new in the field. The company is Finnadvance, and present the application of the AKITA platform in the immuno-oncology. The AKITA platform is manufactured by a company called Finnadvance, or well, with such a name, based in Finland. We're 6% based on site. And most of the technologies that I will be talking to you about rely on the microfluidic plate that you can see here on the right-hand side. In a nutshell, the way that we focus our activities is to go with medium and high throughput. So we keep focusing only on standard well plate format. Those are 96 well plates, 34 well plates coming soon, and having between 32 and 128 tests that may be performed separately. And this capacity elicits us to go with different cell culture options on the left-hand side, which is about the co-culture of cells, organoid tissue cultures, vascularization of epithelium, which lead us to develop internally immunocompetent model for immuno-oncology, assay-ready kit, including a fast-maturing CACO2 vascularization, vascularized epithelium, including gut, blood-brain barrier on chip, and as well to study vascular disorders. The way that the platform is working is as following. So we do have 3 wells that are linked according to a specific microfluidic pattern. We do have the bottom microchannel in red that is in contact with the top channel in blue and separated by a semi-permeable membrane. This is similarly like a transwell. This membrane, usually PET with different pore size, can work as well with different biodegradable or biocompatible scaffold. And the way that is usually done is to have the tissue of interest on the top cells, organoids, what is of interest, and beneath the membrane, another type of adherent cells, whether this may be endothelial cells or macrophage, depending on the model. We can also have PBMC or additional non-adherent cells that are into the microfluidic, into the microchannel, and in which we're doing a bilateral flow, mostly through a rocker. I would like to highlight that despite we're a microfluidic company, we work both into static, where our plate can be friendly for the co-cultures on both sides of the membrane, because we can flip the plate upside down anytime. We work as well with bidirectional flow, and those have been like the most used into the studies that I will show you later. The second is used with a rocker. And additionally, we do have as well adapters in order to plug some pump for very specific application. With such feature, we'd like to position ourselves as easy to use. We're compatible with pipetting automation, microscopy automation as well. And I'd like to highlight that we're also full plastic for low drug absorption and as well to high microscopy performances. To the best of our knowledge, we may be one of the highest throughput on the market, with our 384 well space coming soon up to 128 tests. And we aim at positioning as well as something that is affordable, a kind of standard lab consumable with a pricing order of magnitude below €200 per 96 well plate. Having said that, this is the single organ-on-chip platform that we're able to do. The flow is mostly generated with a rocker. We do have the capacity on the right-hand side to work with multi-organ-on-a-chip link with a single microchannel. We do have our own tier platforms in order to study potential toxic effect, potential leakage into the epithelium, and very importantly, our goal is always to be compatible with what is existing. So we do have adapters for pump upon need, or as well for the low manual tiers for specific applications. Here, we may have different advantages, and this is very tissue dependent, application dependent. This is just a very broad image. Working statics, this is mostly of interest for co-cultures, but we could show, for example, working with CACO2 that we've been able to decrease the maturation time by more than half, can improve the tightness into blood-brain barrier, or the polarization into A549 lung-on-a-chip model. And importantly, what I will be talking about to you today is about the in vitro data we obtained into the oncology field. This shows a bit this different area, which has been a summary, and now I would like to deep into the focus of today, into the oncology with 3 different case studies. The first one is really focused about the drug resistance on a chip. Since we know that chemotherapy is facing major drug resistance, it's important being able to predict it into the patient. And we first wonder what was the influence of vascularization and flow on such a specific phenotype. Into this setup, we've been working with patient-derived pancreas organoids, having as well endothelial cells at the bottom and bilateral flow. We use the standard of care gemcitabine chemotherapy and have been visualizing the tumor survival through live dead cell imaging. Here, in a nutshell, using a patient which we know is sensitive for this drug, having the setup into the static, into the flow, or into the flow with the endothelial cells have very minor influence on the drug resistance pattern. And this, our collaborators from Helsinki University into the HiCA lab then have been simplifying the setup. So getting only the pancreas cancer organoids, plus minus flow, but this time as well choosing multiple patients. The number 3, which has been shown before, has been sensitive with no recurrence in pancreas. Patient 1 is sensitive, but in red, it is resistant. And checking into a no-flow setup, when gemcitabine dose response, we see exactly the same behavior between the 3 patients. However, when you turn on the flow, we could clearly restore the drug resistance patterns that has been observed into the patient 2. That indeed shows that the flow in the AKITA plate potentials the drug resistance in the pancreatic cancer. Very interestingly here, and there are additional study ongoing, is that into these patients 3, there has been a recurrence, but only in the lung. But we don't know yet whether it's due to metastasis or a new cancer, and this is something currently investigated in further studies with higher patient number. We have been as well wondering, why do we see these drug resistance patterns specifically upon flow? So what are the cellular pathways that are involved here? And we've been carrying here the patient's single-cell RNA-seq, and overall having an idea about the multiple pathway involved that you can see here. We could observe an increased metabolism that has been shown indeed to be implicated into drug resistance pattern. We have been as well showing an inhibition of tumor suppressive pathway, whether it's p53, we could see as well an inhibited apoptosis. And finally, a very strong inhibition of multiple inflammatory pathway, including TNF-alpha, interferon gamma, or TGF-beta. There is additional studies that is currently ongoing, and this work should be published very soon. And also, we're currently doing as well some tests for personalized medicine possibilities. But in addition as well to the work that has been shown previously, it highlighted that the immune response is very important in such a model. And that's why we've been putting as well immune cells into the system. We have been a very rapid proof of concept to compare how does it behaves compared with the state-of-the-art transwell. So showing CXCL10 gradients in order to have PBMC migrating through. And we show that at 3 hours time point, whether a dose dependence of CXCL10 has been performing lower in terms of migrating immune cells compared to the AKITA plate at the same time points. Amount of immune cells as well in the AKITA plates have been increasing over 15 hours, showing indeed a competency of our plate into this system. The interest is, of course, to have the cancer. So we've been here plating the renal carcinoma organoid that's been done by the Grönholm laboratory with the University of Helsinki, and showing that showing an infection with an oncolytic virus that has been inducing the expression of CXCL9 or CXCL10 to trigger the migration of PBMCs. It shows indeed that over time, the RFP reporters into the virus has been very efficient, showing an important infection of the cancer cells. And as well over time, upon infection only, showing a high migration of the immune cells that have been labeled with cell trackers. Furthermore, exactly the same experiment has been carried combining the oncolytic virus, the PBMC, with multiple cell lines. So there is this 7860, which is a very known cell line, and as well multiple patient-derived organoids, number 1, number 2, and 3. And over all those different samples, we have seen that over time and upon infection with the oncolytic virus, the recruitment of the of the immune cells into the AKITA plate has been very important, targeting indeed the tumor. And finally, have shown that migration of immune cells has been very important, but cancer metastasis is also known to be a high hallmark and also deadly outcome. I'll go very quick because I see the time is running. And we do have the capacity to create a kind of niche, having a tube of cells, and in that case it was performed with osteoblast niche that you can see here into red, and we've been tracking the engraftment of prostate cancer cells that are here labeled into green. Very quickly, into a nutshell, by quantification with microscopy, we could see very similar behavior of the mutated tumor cells compared to the mice results. This has been published studies in collaboration with the University of Oulu and the Medinet laboratory. I see that the time is going probably quite short, so I would like to go quite briefly under these published studies, but as well with validation with the standard of care docetaxel, which has been showing a very similar response into the mice versus in vitro, and shows as well a good capacity for plates to predict mice test and hence reduce animal experimentation. With that, I would like to thank our partner, both from the Helsinki University, from the Oulu University, and as well you for your attention. I'm happy to answer any question.
Thank you so much, Sebastian. We do have time for one question, and we have one in the Q&A. The question is, are the effects you're seeing a result of flow or rocking of cells in the well? Any similar information on unidirectional flow versus rocking?
That's a very great question. I think the best control here will be to get a pump system in order to have a control, which we haven't been doing in these studies. I'd like to highlight here as well that the size of the organoid was very low, around like 50 micrometers, embedded into matricel. So it has not been free floating, hence not moving into the well. So I would not expect any slow mechanical force to have an effect here, but having a proper control will be the best case here. Thank you for the question.
I'd like just to first thank you for the opportunity to present here today. Yeah, I guess it's not a surprise for this audience that many human complex diseases still lack effective treatment options. In part, this is caused by the lack of predictivity of simple in vitro models, but many times also in vivo models. This contributes to the lack, to the failure of many clinical trials and possibly for the missing to identify effective drug candidates. At Mimetas, we aim to create or to recreate human disease complexity in vitro and contribute with these potentially more relevant models to drug development programs, as well as to the reduction and replacement of the animal models. Before I continue, I'd like just to show how our system works. At Mimetas, we are focused on the development of the on-chip based models. And for developing these models, we use our platform, what we call organoplate. As you can see here, it is based on a 384 wells plate, and the bottom plate, the bottom of the plate is modified, where we have our microfluidic chips between two glass plates. I'd like just to briefly show how we create our cultures here. Represented in this video is a Trilane chip, but we we have a range of different chip designs, and then of course, and the culture setups. So all the selection of the chip is really dependent on the needs of the model. But I'd like to just mention that all our chips is based on the same technology, that is the phase guide technology, this little rim between the different channels. that allow for the controlled release of the reagents into the chip. As you can see here, the phase guide works as a fluid pressure barrier and will prevent that the liquids overflow. Let's say when you are loading material in one channel, it prevents that it overflows to the other, to the channel next to it. Once all the reagents is included in the chip, these cultures are placed on a rock platform and allowed to grow under flow conditions. Here is just a setup that we normally use as an example to generate tubular cultures. So as also many people before me here today discussed, tumors are very complex entities, and in our models we try to incorporate associated tumor microenvironments in our models, and also to visualize and quantify relevant aspects of the tumor models, like tumor growth, but also interactions with the supporting stroma, and also how immune cells infiltrate and potentially function in these microenvironments. So for the seminar of today, I actually selected a few different models just as examples on how we are creating these models, and also some applications that we have been working on. I will start with the PDX on chip. This was a project where we used prostate cancer cell line, but also material coming from prostate cancer PDX models. We generated optimized the protocol for generating aggregates from this PDX tumor, and cultured this in the trilane chip along with a vascular compartment and also isostromal compartments. As you can see here, these cultures, the cells grow over time, and they are nicely alive. These cultures can be exposed to different treatments, as the example of this prostate cancer cell line that was also included in this study. This was a docetaxel naive cell line, so we can nicely see that it still responds to docetaxel and cabazitaxel, but it is unresponsive to enzalutamide and mitosantron. Next, I wanted to discuss our a few setups we use to study T-cell recruitment and killing in our system. So a while ago, we first published this T-cell recruitment assay. Our initial idea here was to have an assay that we could follow T-cell extravasation towards a chemoattractant gradient. So for this, we also used the trilane, where we used the vascular compartments next to an ECM, and on the third channel here, we added a chemoattractant. These cells were loaded in the vascular compartment and allowed to migrate towards CXCL12. This was very interesting because just with this simple setup, we could visualize already many difference between unstimulated and stimulated T cells. So you can see that the stimulated T cells migrate in a bigger number, even in absence of the chemoattractant. We also saw that there is a difference if the endothelial compartment is initially primed with TNF alpha, which is supposed to increase the addition of ICAM expression and addition of these T cells to the wall of the endothelial compartment. And we also see that there is a difference, let's say, between if we have a chemoattractant or not in the system. So we wanted further to develop this to include a tumor compartment and be able to study infiltration of T cells, but also to use these models for CAR-T efficacy studies. So in order to create this cell culture system, we actually adapted this initial T cell recruitment assay. And again, we used here an endothelial compartment, but we wanted to have the tumor cells slightly closer to the vascular compartment. But we wanted that the vascular compartment was, let's say, allowed to fully develop and be very stable for this assay. So our team developed a protocol for luminizing the ECM compartment. So this enabled us to create a vascular compartment, and later on, when the endothelial cells, the vascular compartment was fully developed, we could include the tumor cells in this luminized space here. And then we can nicely follow how the cells are growing. We can see that they are growing in 3D. We can quantify, we can nicely quantify this and compare these different chips and different plates. We can see that it's quite nicely reproducible. So we went on to evaluate the efficacy of an EpiCam targeting CAR-T cell in this system. So for this study, we selected a non-target cancer cell and a target cancer cell. We can see here that the non-target cells are not really targeted by the EpiCam CAR-T cells. There is no difference in the response to the CAR-T cells compared to T cells. And when we look into the response of the target cells, we can see indeed that these cells continue growing in presence of T cells, but the CAR-T cells are nicely killing these tumor cells, and we can see an increase in this killing over time. And also the presence of CXCL12 in the system improves or increases the killing. This is likely because maybe a higher number of CAR-T cells are reaching the tumor compartment in these conditions. So we can also use the same system to evaluate different tumor and CAR-T cell ratios. We can see that in higher ratios, these cells are already killing tumor cells at 48 hours. We can see a quite convincing effect, but lower ratios also results in quite well killing, or at least it prevents that the tumor cell compartment further grows. So using this system, we can nicely see some kind of dose response to these different CAR-T cell, CAR-T tumor cell ratios. The last part of my presentation will focus on the vascularized tumor cultures. So actually for this culture to use a different chip design is also, we also have 3 compartments here, but we have a larger compartment that allows for the formation of a microvascular bed, but also for the culture of bigger, let's say tissues or micro tissues like spheroids, organoids, or even small tissue explants. So we have a very nice, we can generate a very nice and perfusable vascular bed, as you can see here, by use of the FITS labeled dextran. And the idea of this specific experiment was to deliver T cells into a tumor compartment using this system. So we generated the vascular beds, we made a monolayer of tumor cells on top of it, and loaded the system with CAR-T cells. We can nicely quantify here that there is some migration in both conditions, if the tumor cells are present or not, but the recruitment of CAR-T cells, or the amount of, T cells that reach the tumor compartment, is increased in presence of a chemoattractant, and also the conditions where we have also priming of the endothelium let's say, makes the T-cell recruitment into the tumor compartment more efficient. The last part of my presentation will focus on well, it's a similar system, but here we use the dissociated tumor tissue. So this was a project focused on modeling hepatocellular carcinoma. These cultures, to generate these vascularized cultures, we actually loaded the dissociated tumor together with calves and endothelial cells. These are loaded in an ECM and allowed to reorganize. And then we can see here how this vascular bed it's very nice organized, and also that the tumor cells are nicely growing here around the vasculature. So this system, these cultures were exposed to several treatments, as we can see here, and we can nicely pick up a response to specific compounds, but we can also pick up different response between the different patients. Among these different treatments, we actually also included two treatments that are used as the standard of care for hepatocellular carcinoma. So we treated these cultures with sorafenib and levantinib. And these compounds are known for decreased tumor vascularity. So we wanted to know if we could pick up that in our system as well. And then we can see that both treatments, they disrupt the organization of the vascular bed. And we are actually interested in to evaluate if the other compounds, I must say that most compounds included in those treatments that we used were targeting the supporting stroma. So you also wanted to evaluate if the other treatments would also disrupt these vascular beds. And the Now Data team, they developed a tool.
Carla, I just wanted to give you about a 1 minute timing notice.
Okay. Yeah, I'll finish up. So we, using CD31 stainings, we could extract 15 different descriptors to characterize the organization of these vascular beds. And then we analyze this data. We can see that only indeed sorafenib and levantinib are able to disrupt in a similar way these vascular beds. We can see this by PCA analysis of Disney visualization. And just to conclude, I'd like just to emphasize the contribution of on-chip systems for generating very advanced models that allow for the interaction of different cell types and potentially capturing in vivo biology. And hopefully this will contribute for an efficient evaluation of the various therapeutic modalities, and contribute also to the adoption of the MPS platforms, and also facilitate the development of novel therapeutics. Thank you.
Thank you so much, Carla.
So good day, everyone. I just want to start off by thanking Lauren for giving me the opportunity to speak today. And I'm going to go over some of the research tools that StemCell Technologies has developed for the culture of human epithelial tumor organoids. So next slide, please. Great. So StemCell, we've worked in the organoid space now for over a decade. And during that time, we have commercialized so far 12 culture kits for growing different types of human epithelial cells. And I've shown them here. We also have quite a few kits still in development and coming out the next year or so. So if you have any interest in these, check out our website at stemcell.com. So next slide, please. Okay, so I'm going to talk about 3 research tools. One is our PancreCult organoid growth medium, which is used for growing both normal and malignant human pancreatic organoids. We have our InSphero organoid growth medium, which is a kit used for growing both normal and malignant colon organoids. And we actually have a new type of culture plate specifically designed for organoids. So I'll go over that. So next slide, please. Okay, so my screen a bit here. Okay, so the right side of the screen is a little bit cut off, at least on my end. But Lauren, can you shrink it down a little bit? I'm not sure if it's just my end.
Yes.
But anyways, I'll continue. So we have developed PancreaCult. It was developed by Nina Quiskamp, who's a senior scientist here at StemCell. And it's can be used for growing both normal and tumor pancreatic organoids. So the top left graph shows the growth of normal pancreatic organoids in our in-house formulation versus some published formulations, and ours seems to work pretty good. And in total, she's actually tested a total of 5 donors when she developed this media. It also works very well for growing a variety of pre-established organoid lines. And as well, we managed to get hold of a primary tumor sample, and it worked well for that. And that's shown on the bottom left. Now, one potential problem, and it's a recurring problem in cancer research, is that when you get a biopsy sample from the operating room, that biopsy sample often contains contaminating normal cells as well as tumor cells. And when problem that emerges when you put them in culture, the normal cells tend to proliferate and take over the cultures. So when Nina made this kit, she made it as a modular kit, and she purposely packaged the EGF as a separate supplement. Now, vast majority, like over 90% of pancreatic tumors have mutations in KRAS, so they don't need EGF to grow. So if you want to grow normal organoids, you can include EGF and they'll grow great. And if you want to get a pure tumor cell culture, then you leave the EGF out. Next slide, please. And so Nina did some drug testing in some 24-well plates using a couple of organoid lines and tested 2 different drugs, irinotecan and 5-FU. And you can generate nice dose response curves and calculate IC50 values. So we think this could be a useful model for pancreatic cancer research community. So next slide, please. Okay. We have also developed ATCC. It's a complete kit for growing normal colon organoids. Now, again, when this kit was made, it was made to be modular. Again, so we have a supplement which contains your Wnt stimulants, and then the basal component, which contains all the other necessary components required for organoid growth. And again, it's the same problem. When you get a biopsy sample, you could have contaminating normal cells in there. But since, majority, not all, but certainly a large majority of colon tumors have mutations in Wnt signaling, you don't need to actually have to have Wnt in the media to get tumor organoids to grow. So again, and if you're dealing with a type of organoid, a tumor sample that has doesn't have mutations in the Wnt, then you'd have to use the complete medium. But anyways, so just using the basal component of this kit, Martin was able to take 4 tumor organoid lines and grow them up in culture. They grow great. They each have their own mildly distinct morphologies with varying degrees of differentiation. And if you go to the next slide, please. And again, you can do a variety of drug screening on healthy organoids and colorectal cancer organoids, and you can identify which drugs are successful in inhibiting tumor organoid growth and which ones actually have no effect. So next slide, please. Now, one problem with the organoid system is that it's largely a purely epithelial cell system. And of course, if we want to replace the use of animals in research, we want to increase the complexity of the system. And given the success of immunotherapy or the emerging success of immunotherapy in treating cancer, of course, we want to now introduce immune cells into the culture system. And we think that would be a very valuable model. So I'm going to present just a little bit of data here. It was a collaboration between StemCell, and Martin was the lead scientist on this, and the hub organoids in the Netherlands. And what I'm presenting here is more of a case study. It was just done on a single sample as a proof of principle, but it worked quite well. So anyway, so they picked a colorectal tumor. They dissociated it to single cell suspension. They pulled out of that the epithelial cells and established a patient-derived organoid line. And then they took the remaining tumor-infiltrating lymphocytes and expanded them up. And then if you go to the next slide, please. So what we want to do is we want to do a co-culture system, but we weren't actually quite sure what medium to use. So StemCell, we have a T-cell expansion medium called Immunoccult XF, and it works really great for expanding T-cells. And we also have our Intesticult kit. We're using the basal component without the wind, and it works great for growing the tumor organoids. But we weren't sure which one media we should use, or if we should use a blend. And it turns out actually it's a blend, actually works quite well. So if you go to the next slide, please. And you can see that here on the left. So on the left-hand graph here, this is organoid viability, so just organoids alone, in variety of media, so over 5 days. So the blue line is organoids in 100% organoid medium, and the black line is in T-cell medium. And obviously the organoids are not that happy in the T-cell medium, but you know, about a 60-40 blend works okay. If you look at T-cell viability, in the different media. So the graph on the right now, you know, what we found is actually, well, T cells don't like organoid media, and surprisingly, they actually did the best, not in pure T cell medium, but actually in a approximate 1-to-1 blend. So if we go to the next slide, please. The next thing they did is they did a co-culture where they grew T cells and immune cells in co-culture and looked at T cell activation and using interferon gamma secretion as a surrogate. And again, you can see on the left-hand graph that a 50-50 blend, you know, works pretty good in a T cell activation. And the right-hand graph is just a variety of controls, and they all looked fine. Next slide, please. So then when you set it up as a co-culture and you image it, so if you culture the patient-derived organoids and the lymphocytes together, and then you look at apoptosis in the culture through caspase 3 and 7 activation, you can see there's lots of green killing. So that's on the left-hand side, and it's quantified on the graph on the right. If you just do the patient-derived cells only without the T cells, of course, there's no killing. So it's indicating there's a certain degree of specificity and it's T cell mediated. And if you just grow organoids and you treat with staurosporine, which is it's positive control, it's a caspase 3/7 activator, again, you get lots of killing. So next slide, please. So we think that this a good model. It demonstrates that by using a 1-to-1 blend of our Immunoccult and our Intesticult, you can maintain co-cultures of both cell types. And the T cells are active in these conditions. And overall, we think it's actually a pretty good little model for studying T cell tumor cell interactions. Okay, and the next slide, please. Okay, I'm going to change gears here and talk about a new type of tissue culture plate that we have developed, which I think people might find useful. This was developed by Michael Hyatt, who's a senior scientist here on the bioengineering team at StemCell. Next slide, please. So typically, when we do organoids, you take you embed your cells of interest in a droplet of matrix gel, typically 20, 30 microliters or so. And you drop it in the center of a well. We typically use 24 well plates here at StemCell. And then you overlay your droplet with some media. And if you look at the bright field image on the right, on the top, you can see there's a bit of a problem because the meniscus of that media when you look at the cells under a bright field microscope, it makes it hard to image things. And sometimes we screw up and we don't put the droplet in the very center of the dish. It's actually off to the side, and then it's hard to image things. One other problem is with this is that because the dome has quite an arched architecture, you have to use the matrix gel fairly concentrated because if you don't, the gels get kind of wobbly and they break apart if you do a media change, or sometimes you just wash the whole dome away and they get sucked up on a media change. So if you're using matrix gel at a higher concentration, and particularly if you have larger domes, if you go up to say 40 or 50 microliters, you start having a bit of a dead zone in the center of the matrix gel droplet. And you can see that here on the right, on the bottom right. These are some mouse prostate organoids I grew, and in straight 100% matrix gel. And as you can see, the organoids at the periphery nearest the media, they grow nice and big. But as you work to the bottom left of the image and you go to the center of the dome, they get smaller and smaller. So again, because they're not getting the nutrients. So next slide, please. So to get around this, Michael designed a new type of plate, and it's a series of steps in a well. The plates come in a 24 and 96 well format. And so if you want to do an organoid culture, you can put your matrix, say matrix gel, with your embedded cells in the very bottom step there, and then you fill up the rest of the well with media. If you have very big organoids, large branching ones, say a mammary one, you may want to use matrix gel in the bottom 2 steps, and then you add your media. But note that when you add the media, you just add it to the top ledge of the highest step, and you make it flush. And because of that, you don't have a meniscus, and therefore the imaging is much cleaner. Go to the next slide, please. And now one thing is, because we're not putting matrix gels now down as domes, you don't have that arched architecture. It means that you can actually dilute your matrix gel out more. And matrix gel, I know I buy a lot of it, and it's horrifically expensive. So using these organoid plates, you can say, for example, when we cultured intestinal organoids, as shown in this example here, you could dilute out your matrix gel 5-fold to just 20% solution, and you can still maintain structural integrity. Whereas if you're doing a dome culture, anything beyond a 50% dilution, the domes start losing their integrity. So a little bit of cost saving there as well. Next slide, please. Okay, so we've done a growth comparison, again, using intestinal organoids in conventional dome culture versus the organoid culture plate. And they both grow well with essential identical growth rates. And you can see that on the graph on the left. But one thing we did notice is that when you grow your organoids in the organoid culture plate, they're more uniform in size. And again, you don't have that zone in the center, maybe, where organoids might be smaller or whatever. So we find there's a reduced variability in the organoid size as assessed by cross-sectional area. So if you just advance one more slide, please. So overall, we think these organoid plates will be very helpful for the field. Again, so one thing about these plates is that your organoids are always in the same spot in the plate. So if you're doing like imaging, it's easier to track a single organoid, and the organoids, if you're have an automated imaging program, it's it's always in the same part of the plate. So that's really good. Again, there's less meniscus, so you get better imaging, more consistent organoid sizes. And if you just think about like a high throughput applications of a robot, we think a robot can handle these types of plates a lot better than say a regular tissue culture plate. So I'll stop there, but I welcome any questions that you have. Thank you.
I'm here to talk to you today about all the translational solutions that we at InSphero are developing in oncology and immuno-oncology. I will not be able to cover the breadth of solutions that we have, especially with our primary tumor models, but I will give you a little bit of a teaser with the hope that you will visit our websites to sort of look at the entire suite of our solutions. When we started thinking about developing an oncology pipeline, I think one of the first things that we wanted to do at InSphero was really understand the gaps in current available preclinical oncology models. And everyone here is, no stranger to these concepts, right? Because I think when it comes to new approach methodologies or 3D in vitro models in oncology, the bar is very, very high because of the available preclinical models that exist. So any new model that is developed has to address an unmet need that exists in the current models. Some of these are really that the models are not really relevant to the disease at hand. So ensuring that all preclinical models accurately mimic human tumor biology is crucial. And we all know that that's not necessarily the case with all the models that we have at hand right now. The ability to mimic genetic diversity, again, a huge problem because we don't have models that can actually give you that patient-to-patient diversity. Tumors often develop drug-resistant therapies over time, and this is a very, very hard aspect to model accurately in preclinical settings. The tumor microenvironment as well, including the immune responses and interactions with other cells, is extremely complex and not fully replicated in many models. And I'm sure the many, many, many mice that most of us have sacrificed over a period of years is testament to the fact that we still haven't been able to accurately replicate the tumor microenvironment in preclinical models. Something that I'll be talking about a lot today is recapitulating of the tumor microenvironment and scalability and reproducibility. I think this is significantly important, being able to have as robust, reproducible, and scalable models to give you that ability to trust your own data, to be able to develop dose response curves in multiple dimensions that will give you much more information about your tumor model is key really to being able to increase the adoption of these 3D in vitro models in oncology research. And I think the other big aspect of it is that most preclinical oncology models today suffer from translational limitations. So there's a huge gap, which leads to attrition in drug development between the 3D model that exists and then the clinical aspect of the disease. So being able to bridge this gap between the preclinical model and the clinical setting becomes key. And that's also something that we've sort of kept in mind while developing the human relevant 3D models. So before sort of jumping into our models, I'd like to sort of give you a sense of how does our 3D Insight spheroid platform differ from all the other sort of 3D capabilities that you have in the market, right? And I'm not here to tell you that one is better than the other. It's more about what your need is and how you compare across different MPS platforms that exist, depending on what you're looking to get out of it. Depending on, what your platform is, whether you're very early in drug stage discovery or you're looking at a validation aspect of things, you might be looking for scalability, reproducibility, the ability to screen in high throughput, where automation compatibility becomes extremely important. Multiplexing endpoints to get the most information out of a well for automation compatibility is also extremely important. And then reducing cost factor per well, again, very important. I think in the context of all of these, especially in early discovery, being able to use a reproducible, scalable model like spheroids is extremely important. In this sort of day and age of generating data that can be used to develop predictive algorithms with AI coming of age, I think the ability to use everything in throughput and develop that data that can be used to develop predictive models has become more and more important. The FDA Modernization Act 2.0 now definitely allows for more widespread utilization and adoption of MPS in drug discovery. So the more relevant we can make these models, the more we're allowing for the de-risking of clinical candidates as it goes into the clinical phases. So when you partner with InSphero for our oncology and immuno-oncology models, where are you partnering with us in our drug discovery platform, and what do you get out of it? I'm not going to go into, the minutia here, but I'll sort of broadly cover the areas that you would partner with us with. So in early discovery, as I mentioned, earlier on, if you're doing high throughput models screening, you'd partner with us to develop the right model for your assay development, and I'll speak to that in a little bit. If you're doing high throughput testing of compounds, whether you're doing a primary screen or you're doing a more complicated secondary cherry picking screen, that's something that we can develop models for you for. Any sort of target identification and validation studies where you're testing different modalities in physiologically relevant models to identify the same target. Again, something that we could partner with you for. Either you bring the model to us or we develop the model for you and miniaturize it for you as well. This is something that we do a lot of, which leads to lead identifications for many companies that are working with either traditional medicinal chemistry platforms or those that are working with in silico platforms as well. The other aspect that a lot of other companies are not able to sort of don't focus on is the therapeutic window. So we do have our twin system where we're able to identify the therapeutic window using both a disease model as well as a normal tissue model, and I'll touch upon it today, which gives you a sense of what that systemic toxicity is going to be for any oncology candidate that you're looking at. And moving on to the more translational aspects of it, I think the ability of our system to identify biomarkers is a huge plus point. We're able to translate in vitro endpoints to biomarkers of response in vivo pharmacology studies to mimic tumor growth inhibition and regression as being predictive. The other thing we're able to sort of develop models for are to translate in vitro endpoints to biomarkers of response in human clinical trials. And this has traditionally been the big question. How do you take in vitro oncology endpoints in an assay, and how do you make it relevant to a human being? And then we have now developed a platform for primary tumor models where we're testing the efficacy in a genetically diverse set of patient primary tissues. And what this really gives you is that intra-tumor, inter-patient variability, because as we all know, tumors are, every patient is different, every tumor is different, and you need to be able to test it in a representative population of patients. And then going on to phase 1B or phase 2 in a clinical trial, our solutions are well positioned to be able to stratify patients as per their on-target PD modulation response. So this now here is sort of an overarching glimpse at all of our assays, the physiological features that we can model, and the endpoints and applications of these physiological features. As I said before, we can do this in either 96 well plates or 384 well plates, depending on the complexity of the assays. What we're measuring, we can do either tumor cell line models, single cell, multi-cell, add an immune component to it. We could do patient-derived xenograft models, as well as human primary tumor tissue models, all in 96 and 384 well formats. In addition to that, as I mentioned, for our especially for our immune oncology offerings, we do many assays where you're adding matched immune components to be able to mimic that immune microenvironment. The applications and the endpoints are many, and I'd have to say that if I had to pick what I really like about our platform is the fact that it is compatible with so many therapeutic modalities. So we've developed applications for small molecules, therapeutic antibodies, ADCs, engineered cells, oncolytic viruses, and DARPins. And this is a very, very comprehensive set of modalities that you can use to test on our platforms. As always, we do a lot of tumor regression, cell viability. We were able to look at multiple mechanisms of action. And I think the other big aspect of doing something with a scalable model is the ability to multiplex. So whether you're looking at microscopy, whether you're looking at Luminex endpoints, whether you're looking at flow cytometry, the amount of information that you get from a well is tremendous. So what do we mean when we say that we're going to actually develop a model for you as per the biological question that you're asking and the endpoint that you need to measure? So when you come to us at InSphero with a question, whether it's a cell line model, whether you begin with a cell line model and then you go on to validate in a primary tumor tissue model, we would work with you to develop the model as to answer the particular biological question that you would ask. If it's a screening model, we would do this for you in 384, where one of the first things we would do is to measure tumor growth kinetics, viabilities, and then do immunohistochemistry in throughput to make sure that all the sort of biomarkers that we're looking at are actually present in the spheroids. The value for this really is that if you have a tumor or a target of choice, we can develop that screening model with you to test your hypothesis. So you don't have to spend the time characterizing a screening model to answer a specific biological question when you partner with us. The ability to do this sort of takes us 5 to 6 weeks to just develop the model and the sort of the endpoints for you, which you can then use to screen large compound libraries at your facility. As I mentioned, once we've sort of set up the assay, we can run multiple endpoints on it. One of the big asks that we get in multiple formats is for viability. As I mentioned earlier on, we would design the assay as per your biological question. One thing that I sort of cannot emphasize enough is that it is extremely important to understand what biological question you're asking so that we design the assay to answer that biological question. We help you with understanding what the question should answer, what positive controls the assay should have in terms of a biological control, in terms of a technical control. Once you've established the growth kinetics, how long you should take out that assay. And then we do a morphology analysis, and whether you're looking at single or multiple cell type models, we'd either do a bright field analysis or use GFP labeled and RFP labeled cells. So what you can see here is a multi-cell type model where you can follow growth kinetics over a period of time, as well as a cell-based ATP luminescence-based assay where you sort of modulate looking at the effect of different compounds over a period of time. So the value, again, for anyone who's looking at these sort of assays is to use these to develop in vivo relevant insights for tumor growth inhibition as well as tumor regression. So what this ends up doing for a lot of people is this particular assay is used to de-risk in vivo pharmacology studies for a lot of companies going into prior to going into phase 1. In addition to all the all of these assays that we've developed for small molecules, we've also developed an antibody penetration assay. Now, very, very, as you can imagine, antibodies and large molecules are sort of a different beast. And to be able to even understand the efficacy of an antibody, one of the big things that we had to see was in these multi-cell type models and in these primary tumor models, does the antibody even penetrate these tissues? So we developed a 3D inside tumor model where we looked at antibody penetration through confocal imaging over a period of time to determine the best depth of penetration for the antibody. And this differs from antibody to antibody. And as you can see here, we've over a period of between 15 minutes and 4 hours, you can already see a difference in the depth of penetration of this beta integrin antibody. And then over a period of time, we took this out to 24 hours, and you can really see it penetrate the spheroid. Now that is for a beta integrin antibody. For multiple other moieties, you get more or less penetration depending on the tumor and the tissue type. What this really does for a lot of people working with ADCs as well as antibodies is it takes the guesswork out of assessing antibody efficacy. You can test the penetration of an antibody in a primary tumor tissue or a cell line developed model to ensure target localization and engagement prior to assessing efficacy, instead of going into efficacy and then wondering why the antibody was not efficacious.
I just wanted to give you about a 1 minute time warning.
Sure, absolutely. And then in terms of our immuno-oncology testing with 3D Insight, with our 3D Insight models, we've developed a couple different models where we involve the immune system, where we're able to assess immune cell activation by looking at pro-inflammatory cytokines. We have high content imaging ways of looking at phagocytosis. We look at the tumor microenvironment by looking at tumor cell killing and cytokine release. Again, these are very sort of elegant systems that are scalable and reproducible, where you can validate the differential effects of your favorite sort of immuno-oncology therapeutic candidate in a human relevant tumor model. And then, as I mentioned earlier on, we do have this twin safety model, which really helps you understand the therapeutic window between two different tissue types, where you have the tumor type and then you have an organ, a non-diseased organ, and you can look at the effect of the same therapeutic target, the same therapeutic that you're testing, to determine the therapeutic window between the diseased and the non-diseased tissue. So what is our value proposition to you? Why partner with InSphero? What problem are we solving for you? We develop models that represent patient heterogeneity. We develop the model and characterize it for you. We represent the total tumor microenvironment for you, whether you're working with primary tumors or with cell lines, and we give you the most information that you can in a scalable, reproducible, and robust manner. And with that, I'm done.
All right. Well, great. Thank you for the opportunity. Thank you for those who stayed around, and I will be very quick. So what I want to tell you about is what Aracari produces. We have various platforms that we do in-house work, but we also have platforms that we're now shipping to customers so that they can use in-house. We have our vascularized microorgan, or VMO. This is perfused human vasculature on a chip. We have the VMT, which is the vasculature with different kinds of tumors around it, and pretty much every tumor we've tried works, as I'll show you. And then we have two products that we now ship to customers. The Go chip is the VMO shipped to your lab. That's for DIVI studies, drug-induced vascular injury, anything to do with leukocyte delivery to tissues, etc. And we've just started shipping a Go chip Duo, and I'll go through that. And that's a vasculature to which you can add your own tissues in your own lab. So this is our technology. Like everyone, we're trying to hit this sweet spot between simple, non-physiologic, and physiologic and not very simple at the other end. So the vascularized microorgan in the middle, we think, gets the best of both worlds. It's a complex tissue, it's fully human, but much more high throughput and probably more relevant than mice in many situations. So it's based on your vasculature, artery, vein, capillaries, connecting those. We schematize it this way. You're probably all familiar with this idea now: microfluidic channels representing artery and vein, capillary network that connects the two. Here's the vascular network, endothelial cells here expressing GFP. There's a complex extracellular matrix. There are stromal cells which are not labeled. Here's an example of those stromal cells. These are pericytes that wrap around the vessel, just as they do in vivo. And all of this has been collapsed down onto a 96-well plate format. And we have 12 individually addressable tissues on each plate. So each column of the 96-well plate is one unit. and you can see how the tissue chambers where the tissues are line up with the individual wells of the 96 well plate. So you can image the tissue. And here is an example of one of our earlier platforms. And you can see the vasculature in red, and this happens to be tumors growing there in green. Again, hopefully you're familiar with why one would want to vascularize tissues. So as we know, all organs are vascularized, cells are very close to vessels, all drugs and nutrients are delivered through vessels, and critically immune cells and metastasis, in many cases, also delivered through the vasculature. So the best way to model these human tissues is to have vascularized and perfused tissues. So this is just an image. The larger vessel is the artery, and you can see the capillaries sprouting off. Flow through this platform is driven by a hydrostatic head. So we don't have pumps, we don't have rockers. It's very simple. It's just gravity-driven flow. And you can see this is actually blood flowing through vessels in our platform. This is blood in skin. You can see it looks very similar. We can calculate the flow rates, the shear, just by doing computational fluid dynamics based on images of the vasculature. And we can do permeability studies with different molecular weight fitsidextran. And you can see the larger ones, the size of antibodies, leak slowly. We can calculate permeability coefficients, and those match what you see in vivo. And we can challenge the vasculature here with LPS and induce a leak. So it's a fully physiologic vascular system. There is a complex matrix surrounding this, so we like to mimic the real tissue. There are fibroblasts in there, and these make extracellular matrix proteins, and we've listed some of them here. We also get remodeling of the matrix as collagen 1 is laid down, and the vasculature lays down a collagen 4 rich basement membrane. And again, you can see the pericytes that wrap around these vessels. So it is important to note that all the flow in our tissues is through blood vessels. There's no leak. The only way that nutrients get to cells is through the vessels. So it's very physiologic. And indeed, all of our platforms are tested before we do any experiments to make sure that there's flow and that there's zero leak. If there's any leak from the vessels beyond a physiologic level, then those tissues are not used. So you get more accurate physiologic data. We've tested multiple tumor types. Everything we've tested grows in the platform. So here we've got examples of colon tumors, triple negative breast cancer, glioblastoma. Interestingly, as you can see, the glioblastoma cells wrap very close to the vessels, so they seem to really like this vascular niche. And the bottom right is a new platform that I'll talk about a little bit, where we've got this second chamber, which is representing the peritoneum. We've lined that with peritoneal mesothelium and then added tumor cells to that. And so this is a model of peritoneal carcinomatosis. You have the vasculature underlying the tissue, the mesothelial cells, and then the tumor cells are light onto that mesothelial surface and then are fed by the underlying vasculature. We've also done patient-derived tumors in the platform, and these data are published. We've done multiple patients. Some of the tumors grow slowly. Some of them are highly invasive. We've done drug studies and compared these to patient outcomes. The top two, you can see, were resistant to Folfox in orange. And indeed, this patient progressed on Folfox and died. This patient, P61, you can see was responsive to Folfox, and we can see that in our platform. And we've tested some other drugs as well to see if tumors that might be resistant to standard of care Folfox are actually sensitive to other drugs. And here they were sensitive to this drug galenicidib. The platform is ideally suited to immuno-oncology because we have vasculature. We can deliver lymphocytes through the vasculature, and in this image, the vessel in red, the tumor in green, you can see lymphocytes have extravasated from the vessel and are now interacting with the tumor. We've tested PBMCs, CAR-Ts, gamma deltas. We can also do antibody drug conjugates and bispecifics. All of these have been successfully evaluated in the platform, and we can do studies on those for customers. Just an example here, this was an EGFR-specific CAR-T directed against EGFR on tumor cells. Here you can see the extravasation of these CAR-Ts over time and blocked by an anti-ICAM antibody. If we put in a control CAR-T, there's no killing of the tumor. If we put in this EGFR-specific, then you can see in the green, this is the CAR-Ts are nicely killing the tumor relative to the control. And if we put in an anti-ICAM antibody in purple, which blocked extravasation, we block killing of the tumor. So again, very physiologic delivery of these CAR-Ts to the tumor. This is an example of an antibody drug conjugate. Various doses of the conjugates, 1 and 2. You can see at the highest doses, they kill the tumor quite nicely. The reason we were testing these, our client was concerned that there might be off-target toxicity of these drugs, and that was indeed the case. As you can see, when we looked at the permeability of the vessels, the black bar is the control, the white bar is a positive control, and you can see at the highest doses, highest dose of compound 1 and indeed at all doses of compound 2, we did get toxicity of the vasculature as evidenced by an increase in permeability. So the various models that we have, what we can do with these platforms, we can do direct imaging of cell growth, migration, survival. We can do permeability, obviously vascular tox, I've just shown you that, leukocyte adhesion and extravasation, efficacy, tumor drug sensitivity, obviously, immuno-oncology, I've shown you some images of that. And if you're interested, we can also do in situ immunofluorescence staining, and we can extract tissue for qPCR and single-cell RNA-seq, which has given us some really fascinating insights. And of course, because there's flow through the system, through the vasculature, we can collect the effluent to detect biomarkers, either by ELISA or mass spec or whichever assay you prefer. So a lot of that work was done in-house. Clients would send us drugs, we would test them. But there's a lot of interest in people doing work in their own shop. And so we've developed what we call the Go chip. This is a version of our VMO platform that can be shipped overnight. So the top green image there is the vasculature on the day we shipped, the day of arrival and the day after arrival. So you can see that the vasculature is perfectly intact after overnight shipping. So we can ship this to anywhere in the US. And the Go chip is useful if you want to do DIVI studies, drug delivery, immune cell extravasation, etc. It's the vasculature. And now we've developed what we're calling the Go chip Duo. This has preformed human vasculature, which we would ship to you. And then there's a second compartment that is accessible to the client, and they can add to that whatever they would like. So for example, on the left here, you can line this second channel with cells of your choice. Earlier, I showed you peritoneal mesothelial cells. You could use gut epithelium, lung epithelium. So basically line this channel with any cells that you are interested in. All right. Or you can add a gel and cells to this channel and add organoids or single cells to the channel. And then these tissues are supported by drug or nutrient delivery through the vasculature. So the nutrients come in the artery, through the capillaries and out the vein, and that provides nutrients to the cells and the organoids that are adjacent to that vascular compartment. So it makes for a hugely flexible platform The customer gets the vasculature and they can add whatever they want to that. And again, this is just an example. The peritoneal mesothelium in blue, the tumor cells in red, the vasculature in green. And you can see over time, some of these tumor cells are migrating through the mesothelial layer into the underlying tissue. And this is exactly what happens in peritoneal carcinomatosis. So we are now taking patient samples and testing those in this platform to identify potential drug therapies. So why Aracari? We have vascularized tissues, you get physiologic drug and cell delivery. There's no necrotic core, as can be seen in some spheroids. We've done a lot of single-cell-seq to show that gene expression mirrors that seen in vivo and is very different to what you see in 2D cultures. It's very easy to create custom tissues around the core VMO, very easy to use, it's gravity-driven. We've had it all independently validated as well. And as I say, we can now ship these platforms to our customers. So this is all the stuff you know, why we want microphysiological systems. They're more physiologic, easy to use, also vascularized, so they're very physiologic. We can do patient-derived stuff. And again, we can now ship these to customers. So with that, I'd be happy to take any questions if there's any time. Thank you.
Thanks so much, Chris. Yeah, there are a few questions for you, which if you have time, we can answer.
Absolutely.
The first is, have you tried using spheroids in your VMT model?
Yes, we have, and they go very well, actually. It's interesting. We're getting what we believe are better data by putting in small spheroids, sort of somewhere between 25 and 50 microns. They seem to mimic the nests of tumor cells that you see in a real tumor. So rather than just disperse single cells, which we've done in the past, by adding these small spheroids, it's really mimicking the architecture of an actual tumor. So yes, that actually works well.
Great. The second question I have here is, do drugs, especially biologics, reach the tumor cells only by extravasation, in contrast to passive diffusion through damaged vasculature? And then do you have data to show this?
Yes, and it depends very much on the tumor you use. So we have tumors that seem to support vasculature but don't damage it to any great extent. In that case, any drug delivery is entirely through the sort of physiologic route of either uptake and delivery by pinocytosis or just free diffusion through junctions. So we do see that. And the permeability studies I showed you earlier, 150 kilodalton fitzy-dextran leaks out quite slowly. You can do that with labeled IG. It leaks out, but quite slowly. We also have tumors that disrupt the vasculature and make it quite leaky. There, it's mostly a disruption of junctions. So it's not where we have just destroyed vasculature. It's just very leaky vasculature. We know that because if we put in a drug called razuprotafib, which stabilizes Tie2, that normalizes the vessels, makes them less grossly leaky, but because it improves flow through the tumor, we actually get better delivery of small molecules if we normalize the vasculature. So the point is, yes, it's a very physiologic system. The vasculature responds to tumors in the same way that vasculature does in vivo, and you can actually manipulate the vasculature with stabilizing drugs like razuprotafib and improve drug delivery that way. And those data are actually published from my academic lab, so you can look those up. My name, UCI, and that will get you to those papers. And I think we've also got them on the Aracari website.
Awesome. Okay, so it looks like that concludes our questions for you, Chris. So I just again wanted to thank all of our speakers from today. Appreciate everyone for sticking around to the end. I am going to go ahead and end the meeting now. As a reminder, we will be sending out the recording to anyone who registered for the event, as well as posting it on our YouTube. So feel free to share with anyone who missed it. And we'll also be sharing the presenter's contact information. So if you do have any questions that weren't answered today, you can go ahead and reach out to them. So thank you so much, everyone.
Thanks,
and
Laura.
I hope you have a good rest of your day.